Hello, everybody. How are you guys? Very good. Is everyone getting anxious about opening of all the stores and restaurants? Let me just answer this for a moment, okay?
You are currently the only person in this conference.
Hello, can you hear me, Suganda?
Testing, testing.
Hello, can everybody hear me?
Yes.
Thank you for confirming. Hello, hello. Hello, beautiful people, says Sharon. Happy Friday.
Happy Friday.
So, I am actually just gonna start soon, so we have a couple more minutes. I wanted to thank you all guys for participating in the SWOT analysis. And before we start jumping into the micro piece, I would like to have just a brief review of how is that working. I find it is a reflection of where you are and how you're feeling about your own positions. But what is really important, and I really wanted to highlight, I didn't want to say anything in the conversation there. I actually wanted to take this opportunity and tell you in person, you're not alone. And you have to tap into the resources that are available to you. I know sometimes you feel like you're fighting that battle all by yourself, but you're not alone. So I see a lot of concerns that are coming up, and this is more not related so much to the SWOT analysis than colleague to colleague comment. So anytime you have an opportunity to tap into resources, please do so. Don't be shy to ask. The worst thing is that you can get is a no as an answer, and you're at the square one. But there is always people who want to support you and help you through this journey. So when you're developing your IPAC program, I noticed that most of you are focused on that you see as maybe some of the characteristics that are lacking, or maybe something that you could be improving on. And that's great that you have that insight. But I just don't want you to be discouraged and think that this is a battle that you have to fight by yourself. So if you need anyone to bounce any ideas, I'm happy to talk to you anytime. You can email me and we can set a phone conversation. I'm happy to give you my insight and help you with that. But it's just whenever I was reading any of your posts and as in the SWOT analysis, it was just resonating that you are isolated and that there is a lot of work ahead of you. So I just wanted to highlight that. And then with regards to the SWOT analysis, you did a great job, and I also like how you summarized into kind of smaller, achievable bites. So just something that you can use in the instance that you do need to develop something. It is important that you take that step back and take a look at what do you have and what is it you need moving forward. So that said, I also would like to highlight a couple of challenges that we are facing. We are still having challenges with the videos and all of the presentations being captured and uploaded in timely manner. Um, Leonard has been absolutely amazing helping and doing all the work. So I just don't want you to stress about this. I know it is important for all of us to stay on time, but when it comes to things that we need to do, we will, if it comes to a point that we need to push some of these dates, I'm open to reconsider some of those dates that may affect how are we going to move into the courses and so on and so forth. Anita can't hear me. Can anyone else hear me? Don't worry. Don't absolutely, Justina. Don't worry about that. It's as I have reached to some of you on personally because I was just getting the sense that you're getting really stressed over this course. The course is not here to cause any pain and suffering to any of you. The course is here to help you get to CIC, and I'm going to take you on that journey. I'm going to help you get there where you need. So sorry, Anika. I don't know why is it choppy. Can everyone hear me right, or is just Anika that is having hearing? No, and this is not a goal, Vernette, to get you to a place that you're sweating bullets coming on Friday class. We have to have fun. Look what I'm wearing, and I have my ready for you guys. All right. So did everyone get your water and chocolate and some snacks to get us through this busy presentation tonight?
Oh yeah, I just came in from a walk.
Perfect. Very good. So yeah, this is what I do on occasion. I go a little bit crazy and overboard and just want to make people smile when I'm delivering some of the news. And I also give them sometimes these. I don't know if you can see well. Oh, there you go. So for all my colleagues that do support IPAC and they show that a little bit of extra, I like to give them things like this. So, all right, let's start. So I know we have some of people that are from the micro background, and by all means, I do encourage you to share your knowledge. There is no better way for any one of us to learn than actually share the knowledge among each other. I already have seen that Anita has posted some of the videos, so please go ahead and take a look at those. But as we go through, we will chat and take a look at some of the bits and pieces of microbiology and how that affects us as IPAC in the field. So you will find that there's a lot of information out there that is part of micro, but we really don't get that much involved in. There is certain aspects of micro that we as practitioners need to be very comfortable with. So through this 2 sessions today and tomorrow. We will cover some of the emerging microorganisms, some of the AROs. We will go through, I'll take you through all significant microorganisms, and I will also make a point of highlighting what can come up on your CIC exams from my own experience. So I will just make a little highlight there, okay? All right, ready to rumble. All right, so let me take you back through the history of micro. Hello. Hi, Rosemary. So I'm going to take you a little bit through the history. So the microbiology kind of starts with the development of the microscope, of course, and Mr. Leeuwenhoek was the one who developed the original microscope that actually opened the world of microorganisms to us. So you can see in the right corner, there is a book that he has published. Can you guys see the entire slide, or is it, again, just the mini slides that you see? So let me know if there's problems with seeing the slides. I will adjust and try my best to help with that. Mini slide. Okay, let's see. Is this better? Now someone has to say, because I don't see anymore.
No, the slide is fine. It's just that we have to maximize it full screen to see it better.
Okay,
Yes.
got it.
Yeah.
Okay. All right. So, okay, let me go back. All right. So in the who was it? Carrie. Carrie, can you just maximize? There is like a little 4 arrow in the corner of the slide. If you expand that, you're gonna be having a full slide on. Perfect. So back to Mr. Leeuwenhoek. He actually was the one who developed that microscope. He wrote a book about the little creatures that he saw through these microscopes. So in the 1674, he described the protozoa called the little. Hi, Rosemary. Can you hear us? Did you, Leonard? Yeah, I think he deserves to be reported. But as we go through, there's going to be a little bit more of a juicy story, more of before whatever happened before reality TV. So, and then he also, in the 1683, he for the first time actually has described the shapes of the bacteria. He saw them as rods and spirochetes and cocci. So that's kind of cool and interesting bits and pieces for us. And then there's a bit of my own nightmare. There was an assumption that organisms somehow grow on their own. So Mr. Jean-Baptiste Hamond had an idea that if you leave dirty shirts for 21 days to kind of baste in their own juices and you add some of the wheat, you will have a full-grown mouse in 21 days. Thanks goodness he was wrong. That's not really happening. We are not having any mice growing out of the shirts. But Mr. Georges Louis Comte de Buffon, not that I'm gonna ever say these names ever right. I always butcher all the names. He supported this theory. He actually thought that the organisms do create out of nothing, right? So there is these existing molecules that turn into mold, and then somehow by magic, this creates another fresh organism. Luckily, neither of that is happening because a lot of stuff would be growing around. We are lucky that's not happening. And I'm super lucky that these mice are not growing out of shirts because that's really a nightmare to have. And then Mr. Pasteur, who actually now is trying to contradict some of the other scientists that were operating in his time. There was Mr. Pouchet, who actually what he thought was if you boil the water and boil hay and you put it together, it should not grow anything. However, he was somewhat right. However, when he boiled the water, added boiled hay, it still grew some muck in the bottle. So Pasteur knew that that could not possibly be right. So he was saying that there was a possibility that something actually from the air has contaminated his samples. So Pasteur takes the similar approach, and then he decided to take 2 flasks, boil the meat broth, and one flask was with the open neck, and the other one he actually bent the neck away. So in the one that had unbent neck, that grew again some microorganisms. However, the flask with the bent neck did not grow anything. And of course, now the gloves come off and the gentlemen start fighting. So Pasteur actually had a statement where he said that the doctrine of spontaneous generation, so thank God no mice self-generating, will never recover from the mortal blow inflicted by his experiment. So, ta-da, take that, mice. And then after that, of course, Mr. Robert Koch comes onto the stage. He actually had not had a good relationship with Mr. Pasteur. So they actually had an argument, in-person argument, where Koch has insulted Pasteur, saying he is not a physician, therefore he couldn't possibly bring any value to the assessment and decision making or any of the judgment of the pathological processes. That's way before the housewives of whatever counties enter stage. Okay, those gentlemen misbehaving, calling each other names and insulting each other. But what is really interesting about Mr. Koch is that he also had a fairly similar idea to Pasteur, looking into the development of diseases. So he knew that the pathogen had to be present in said organism in order to transfer to another organism. So he knew that you had to be able to isolate that microorganism out of the, say, another mouse in there, as you can see, and then transfer it into pure culture, and then proving that that actually can grow. So those are your Koch postulates, and out of it, pretty much all our microbiology and all understanding of transmission of diseases is stemming out of it. He has been one of the contributing great grandfathers to our microbiology. So that's a little bit of drama among our microbiologists that we now blindly follow in the 21st century and like to reflect back to their findings. So what are we doing right now? We're dealing with microbiology, which is a study that includes the bacteria, fungi, protozoa, viruses, algae, parasites, and prions. So bacteria, you guys know, they're single-cell organisms. They're prokaryotic. Fungi, those are your molds and yeast. Some are beneficial. We use them to make bread and beer. Others are not so grand to have. Then you get into your protozoas and viruses and algae, parasites. Those can be internal and external, and then prions. So one of the most, I would say, best known prion disease would be the mad cow disease or Creutzfeldt-Jakob disease. So we will be getting into a little bit more detail as we are going through this class. Okay. So when you're looking into bacteria, what is really important for us to know that they're single prokaryotic cells. They do have a single chromosome with no nuclear membrane. They reproduce through asexual reproduction. And they do have unique cell phones, but when you're looking into their asexual reproduction rather, that's done through 3 different ways. It's either conjugation, transformation, or transduction. So I'm gonna, oh no, this is not gonna work. So let me pull my presentation up here. So let me see. I don't know if I can share with you guys. Okay, screen 2. Okay. So that is the bacteria splitting and growing through. I don't know if you managed, did I manage to share that with you? Maybe not.
Sorry, can you show it again? For some reason, I don't know, the light's out in my area. I couldn't see it.
I managed to kick myself out, but I don't know if I managed to share the screen with you guys. Maybe not.
You did originally.
Okay, good. I'm
Yeah,
so
I was saying that for some reason the light's out in my area, so I couldn't see the actual video. So I don't know. So I have to hotspot my phone or something because I can't see anything. Like I can see your slide. I just can't see your video because the connection's very slow.
Okay. Let me try then. So it's just microorganisms. It's actually E. coli growing. Yep, they do duplicate very fast. I apologize for that. Yes, the only problem is that what is running on my screen is actually the presentation that's uploaded into the recording here. So it's, for some reason, I can't open the links here. All right, so the cell envelope is the next topic I want to just bring up to you. So the cell membranes are basically built of phospholipids, and that's going to become important piece of information for us once we get into the staining by gram, because that's what is actually changing the color of the stain. Thank you, Anita. So the main function of the cell envelope is to transport the substance in and out of the cells. So when you're looking into any of the cell envelopes, they're the ones that are these membranes that are letting things in and out of the cell. That will become important information for you to know when we start looking into the resistance of the microbial organisms, and also looking into how are they breaking down the antibiotics. All right. So when we start looking into the way the bacteria is actually identified, going through different ways. One of the names is looking at the name of the family. So when you look at any of the microorganisms at their names, they have kind of like a last name and a first name, if we can say it in that way. So if you look at an example of using Staph aureus, they're always written in italics. Both of their first and last name are written in italics. The Staphylococcus piece in this particular organism is actually what is considered to be a genus. And then when you look into the second term is actually specific epithet of the microorganism. So generally, when you look at the names of the microorganisms, you will see often something like Staphylococcus or Streptococcus, and that is okay. Usually identifies the species, so you will see something like Staph spp or whatever family they belong to. They belong to the species, and that can stand alone. However, aureus alone cannot be anywhere. So we are not on a first name basis with these bugs. All right, so when we move now further into the organisms, I'll also try to give you this as well. There is actually the cell structure is what we are looking at. And sorry for these links, I apologize. So there are small single cell organisms, as I said. They do have a double-stranded DNA. This bacterial chromosome is actually attached to the plasma. So this is a very specific piece of information in relation to CPE microorganisms. So you will, when we get into the breakdowns of and the generation of CPEs and how that knowledge travels from CPE, remember these plasmids are really important in carrying the messages. So they're the ones who carry the genes from one microorganism to the other, and they're not necessarily the same microorganisms. They're kind of remote cousins, like a big family. So you will not need to know all the microorganisms in terms of the structure. What you need to know is when you're looking at your CIC exam, you have to be able to take a step back from the problem they present to you and put your IPAC hat on and then start thinking about what is it, why is this relevant to infection control, how is it transmitted, what's the incubation period, and more around those questions. You will not be asked to identify every microorganism on the planet and have to know all the relationships with humanity.
Sorry, Cornelia, so I wouldn't need to know, like, is a single-celled organism with a long circular molecule, for example?
So
like
you actually, they may ask you about, say, what kind of organism is it? And they will do comparison between, like, say a fungi and viruses, and give you like a question, for example, which one of these is a single cell organism, which one of these requires a host, so things like that. So they will not give you a definitions to recite back to them if that's the question. Okay, got it. So I just thought maybe we can share this. So what's a plasmid? I don't know. Do you guys see this screen? Did you see what I just opened up or not? Probably not. Okay. Oh, Angela, you saw it? Or did you click? Okay. Oh my goodness gracious. I don't know what I'm doing here. Okay, let's try this again.
We cannot hear you.
Cornelia, we probably didn't hear you for like the last 2 minutes. Ever since you tried to play the video, we didn't hear anything you said. So if you narrated, we did not hear anything. Maybe if you exit and try to come back, as long as I think Leonard is the one that is recording it. I don't really know who's recording it, but I mean, if you exit and come back, it might work.
I don't think she can hear us.
No, she can hear us. We can't hear her.
Hello? Oh my gosh, I'm not sharing any more videos.
We can hear you now.
I'm so sorry. Oh my gosh, I apologize.
Cornelia, you know what you did before when you played the video, but then you didn't have to exit. Like we saw the video and you were still talking. If you want, you could just share the video and then just give us a gist of it, or we can just review it later. It's not a big deal.
You know what? I feel like we wasted enough of your time. So let's just keep moving, and then I will stop sharing videos and I will send you all the links that I wanted to share with you. How is that?
Sounds good. We can review it.
Because I feel like I'll just keep wasting your time, and I apologize for that. Oh my gosh. Okay, I will send all the links to you guys. Okay, so when we go back to our bacteria, finally you can hear me. Yeah, I'm all now confused and dazed. So they're broken down into cocci, bacilli, coccobacilli, and I like kind of look of the spiral bacteria. They always look so cute, and so do the Vibriosis spirochetes. So they're always a little bit wavy, and you will find these guys usually in water, so that helps them move along. So for the cocci, you guys know all your streptococci and staphylococci. Yeah, I know, and I'm really not great at this technology. So Leonard must be laughing his head off somewhere in his office. They're thinking, what did just happen here? Okay, and then so with your bacilli, there is your diplobacilli and streptobacilli. Oh my goodness. Okay. I feel like I need to calm down a bit. So when we start looking into our bacteria, as if you remember, I mentioned earlier, the envelope is playing a critical part in the gram stain. And gram stain is actually a methodology that's been developed by Hans Christian Gram. And it was developed in the 1884 and remains in use. So Anita actually shared a video that works on the website. I will upload also some of the videos about the staining and how that works in the lab. That's a link, and I'm not touching it there. Yeah, well, I can't work on one computer, Leonard. You're working on 3. It's just what I needed to hear. So the gram stain is used mostly commonly to help us see the microorganisms under the microscopes. So they're divided into the 2 groups. So you will have your gram positives and gram negatives. So usually when you submit a specimen for testing, and if you're waiting eagerly and calling the lab, they will tell you, oh, we only have so much information right now that we know that it's a gram positive or gram negative. And sometimes they will tell you the preliminary report gram positive cocci in chains or gram positive cocci in clusters. So that will help you a little bit, but you always have to see your final result. So Jagdeep, you're funny. So when we look at our gram negatives, usually you will find them to be stained in kind of reddish color, where your gram positives are usually identified as blue or purplish. So what happens with the gram-negative cells, they only contain few layers of the peptoglycan, and so that's the difference between the gram positives and the gram negative, is actually that peptoglycan that is actually staining. So when you are applying the stains and washing the stains, what remains is the color that is stuck with these the microorganisms. All right, so when you look into the structures of the gram positive and gram negative bacilli, so you will now hopefully see some of the names that you are familiar with. So you have your cocci, that's your Streptococcus and Staphylococcus species. So under the Streptococcus, couple of the that you guys know is your pyogenes group A, Streptococcus pneumoniae, Streptococcus, and then under the Streptococcus there is the Enterococci. Those are your VRE usually. Enterococci fecalis and Enterococci faecium. And then under your staph, there is your staph. Those are usually the ones that you will see with like your staph aureus, staph epidermidis. Both of them, they're on skin, of course. And then when you get into these bacilli, there is one of my favorite microorganisms is actually C. diff, and Clostridium is a bacilli. When you move over to the red bars and red columns there, you will see that they too break down into cocci and bacilli. It doesn't matter whether when you're looking into the shapes, it's really not defining how are they what shape they are when they're identified by the stain. So under the gram negatives, there is your Neisseria, which breaks down into Neisseria gonorrhea and Neisseria meningitis. And then, of course, under the bacilli again, there's E. coli, Salmonella, Shigella, Campylobacter. So what I will tell you, out of these organisms, you have to be very comfortable with E. coli, with C. diff, Salmonella. Some of these will be on your CIC exam. C. diff will definitely be mentioned. Some group A strep may be also mentioned in your CIC exam. Well, we will definitely have it on our exam. Oh, thanks, Anita. Coming from you, that makes me feel so special now. So for the bacteria that doesn't stain by gram, so you will find that in particular when we're talking about Mycobacterium, and Mycobacterium, that's the one that is causing the tuberculosis, they do not stain well by gram. So they're using a different methodology for that, and that's called an acid-fast stain, or it's named after the Zeal Nielsen, who actually has developed this particular technique to stain the microorganisms that do not stain by gram. So as I said, it's used for Mycobacteria, Nocardia, and it's just another way to stain the microorganisms. And we have Mycoplasmas. It's a completely different microorganism, not to be confused with Mycobacterium. They're not visible by gram stain either. So usually to actually identify them, look how cute they are. You actually have to use the light microscopes to see them directly under the microscopes. They can be grown in special enriched broth, and usually that's the methodology that they're using to identify these particular microorganisms. So when you come across this particular microorganism, mostly it's not going to be done in your house laboratory because they do not have the ability to test in smaller labs. And what is also interesting is the duration to identify this one is it can actually be about 21 days, sometimes even longer than that. So capsules, those are the again, part of the makeup of our microorganisms. They're usually polysaccharide, and they do have an interesting function as well. So they do protect from toxic materials in the environment. So they have a protective purpose and protective characteristics for the bacteria, but they also promote the concentration of nutrients around the cells. They do help with adherence. And this is, so just make sure that you do know that the glycocalyx is a part that does adhere to the to other cells. So it is important for you to know that. So some of the examples of those are your Streptococcus pneumonia, Neisseria meningitidis, and Haemophilus influenza. All right, so the endospores. So spores are some of the microorganisms are actually having ability to create these spores. So they could be exo or endospores. When you look at the endospores, so there is an image on the screen for you. That's a Clostridium botulinum, and you see how it has almost looks like a pin, a bowling pin shape. And it's because of the creation of these spores. On the yellowish greenish slide image rather, you can see the location of the spore. So depending on microorganisms, some of these microorganisms will have a more of a center location spore. Some of them will have it to the side. Others will have it on the top or bottom. So what the spore does, and what's really important for us to know in relation, not no one's going to ask you where the spore is, but what's the purpose of the spore, and knowing which organisms are spore forming. So your Clostridium difficile would be one of them. Clostridium botulinum would be again another one. Bacillus anthracis is another one that creates spores. And the spores are used by the microorganisms to survive in an environment that is not the most hospitable environment for them. So usually what you will find is they can survive on the surfaces. Like for example, C. diff. There is different studies that speak about survival of C. diff, but C. diff on a smooth surface, such as your nursing stations and things like that, and say toilet seats and those smooth surfaces that are not cleaned, it can survive up to 3 months. There are some studies that speak about longer periods of survival of spores on soft surfaces such as fabrics, or even there's one study that I came across mentioning 3 years of survival in the fleece. So that gives you a whole different idea about wearing fleece, and maybe mice will start jumping out of those shirts as well. So for you guys that are thinking about how does this actually translate in real life, so when you're dealing with C. diff, it is critical that you know that there is additional cleaning that needs to be done, and only sporocidals can be used to kill the microorganisms. Sorry, I'm just going to take a look here to see what's going on in the chat here, if I need to address anything.
Vernette, I'm seeing your message there. It says next CIC is in September, but they always say that you need like a 1 year full-time or a 2 year part-time experience in IPAC in order to challenge the exam. So that's why I'm trying to ask, like, are we still going to have access to this 1 to 2 years later when we're able to actually challenge the exam?
So I'm not sure how the access to where you are now entering the course works. Maybe Leonard can help us understand that. But you're right. So with regards to your exams, the next exam is actually in July. I don't think you guys will be ready to write in July, considering that our course is running, if you're counting on this course to support you through, the course is running by mid-September. So I think they have the next one, it would be in December or something like that. So it's important for you to check when you can write. I know there are some delays that have happened with writing exams. I know some of my colleagues were not able to write. It was pushed back and pushed back. But so double check on those dates when you can write. So here's an example what would be important for you to know for your CIC exam. It's that comparison between bacteria and viruses. So there are simple cells are the bacteria, where on the opposite side viruses are non-cellular. So bacteria is surrounded by the cell membrane, where the viruses are surrounded by the protein capsid. Bacteria is usually spherical or rod or a spiral, where viruses take more of geometric shapes. Some of them look like screws, and some of them look kind of like almost like some sort of a tennis ball, fuzzy tennis ball, I would say, if that's the best description that I can come up with. Then what are you looking at in terms of diseases, what they're causing? So being able to speak to that. So when you're looking at the way they're also functioning, how does a microorganism, bacteria microorganism, affect the host versus a virus? So just kind of be comfortable with those differences and recognizing that the different organisms that we are dealing with. Oh, that would be so cool, Leonard. Thank you. So, and then another thing is also the treatment. So you will likely have a question around just recognizing a broad information in terms of what kind of antibiotics are used. You will not be asked, oh, what kind of treatment would you be prescribing to somebody that presents to hospital with, say, C. diff, because that's not in our scope of work. But we do have to have a knowledge around some of the antibacterials, antimicrobials. So there is a new section in the book that we will be covering a couple of weeks from now, is antimicrobial stewardship. So that part will be addressing the use of antibiotics. And when you get a question, usually, so what happens sometimes, they will give you a scenario question. I don't know if I've spoken to you about this. So they will give you a scenario question, and then usually can have 3 or 4 following multiple choice questions that are responding to the original scenario. And then they will be asking you about some epi questions in relation to that scenario. It could be occupational health, could be micro, could be also just in around additional precautions and routine practices. All right. So the epidemiological triad, which is something that you have to get a really good understanding and get a good grip around this, because it is important in making any decisions from the IPAC perspective in terms of breaking the chain of transmission. So, okay, so there is an agent, there is an environment and host, and we will take you on a journey through all of these. So with your host, you can have a different relationship between the host and the microorganism. So it could be a symbiotic relationship, could be commensal relationship, could be a mutualism or parasitic relationship. So with symbiotic relationship, neither one benefits or neither one is at risk. However, when you look at your commensal relationship, you will see that there's 2 species that are working together and they coexist, where one of them is actually benefiting and one is at no harm. So this is different between the symbiotic relationship. So in commensal relationship, there is somebody who is actually benefiting from it. And then in mutualism, both are benefiting. So sometimes you will find in nature that, say, a bird is piggybacking on one of those big calves and picking all the flies off the calf, and calf is not bothered by the bird, but they're not having any more the flies on them, and the bird is fed. Everyone's happy there. And then you have a complete opposite relationship there, where it's a parasitic relationship, where the host is the one that is not benefiting, and the parasite is benefiting from this relationship. Oh, yes, Anita, there is actually a stool transplant that is in use for C. diff. And it's usually harvested from a healthy population. And it's quite challenging, actually, to get the healthy stool specimens for the transplants. So it is, it's not really that widely in use as we would like to see it. And it's for the reason that C. diff is not a good competitor. So C. diff is kind of bully in a playground. So if you think about C. diff, when C. diff takes the stage, it's usually after the antibiotic use, it wipes the gut out, and then there is no other microorganisms to fight it over. So it just keeps multiplying. And if you introduce a healthy stool with healthy stool flora, then it's a little bit different story for that C. diff. Okay. So that was a discretion here. Okay, so we are getting now into the relationship between the microbes and humans. So as we mentioned before, there's different relationships. So now you can see what happens when you have colonizing floras and potential pathogens. So with colonizing flora, usually you can think about organisms like Staph aureus, and can usually lives on the skin and doesn't really do anything to us. It's actually part of our skin makeup. In about close to 70% of regular population will have just regular Staph aureus on their skin. So when you're looking into your potential pathogens, those are the organisms that do live on with us and on us. However, given the right circumstances, they will become opportunistic pathogens. So often, I remember from my early years in school when I was taken to the hospital to take a look at the infectious diseases department and the unit. For the first time in my life, I've seen candida in a person's mouth, and it was in one of the HIV patients who was just actually developing the active disease. And candida, that actually is commonly found in our mouth, I was taking over that flora because of his immune system failure. So yeah, you can save your presentations. You can keep that, download them. They're downloadable, and maybe that can help. But there is no recording on them, so not that. I try to put enough information in these presentations that can remind you, and that you can also use them to study as a study material, because there is no other resources for you. And then with pathogens, you can think about, I can think of a few, but I listed in this particular case E. coli, and usually they are part of your normal flora, your normal gut flora. However, as for you guys that work in long-term care facilities and retirement homes, they are very frequent cause of urinary tract infections, and especially in female population or population that does have catheters in situ. So you will find that anything from that feces and the back exit does travel to urinary tract. I try to make that sound less.
Actually, Cornelia, when you're talking about that, I had a question for you.
Yeah.
Well, a lot of the times in long-term care homes and retirement homes, as soon as like a resident exhibits any kind of infection, antibiotics are started. So I was wondering, like, is there, like, is it all these tests that we're doing to figure out if they have something? Because they're saying that if they're asymptomatic, you don't do anything, but they're symptomatic, then you give them antibiotics.
So in particular with urinary tract infections, I can also share a document with you. I was actually involved in a research project moons ago, and we were working with our sister long-term care facility that they were sending us a bunch of their residents, and they were always coming with diagnosis of urinary tract infection where in fact they didn't. So usually they were sending them to hospital with these what they call the bacteriurias, and they're asymptomatic bacteriurias, where you have a urine that's colonized or contaminated due to some of this anatomy of human beings that does things do travel to certain areas. When you're looking into your true urinary tract infections, you're looking into your suprapubic pain. They have to have fever, and of course they have to have a positive urine culture. So we find that quite frequently urine positive cultures are seen as an infection where they're not necessarily a true infection.
Yeah, so I'm seeing like in these homes that they start it before getting it back. And I guess that's part of that whole antibiotic resistance, trying to get everyone education about it. But a lot of the times they start antibiotics before they even get the results because they just isolate them and start something.
Yeah, so and that's work that we need to do, and that's part of your antimicrobial stewardship. We will cover that in week 7, and we're going to get in a little bit more in depth discussing these challenging, and it's something that we just have to deal with because people are set in certain ways. There is a document, I will share that when I can share it after this class or once we get to the week 7. I had a plan, but if you need it, I can send it to you. It's actually a clear definition of what a UTI is. Okay, so I just wrote it down for myself so I don't forget.
Yeah,
so
the broad spectrum it depends on the home. It depends on the home, the doctor, the patient, the history, and everything like that. So it's not always in retirement or long-term care that they just jump and give antibiotics like that.
I think you're right, Vernette. From my experience, I think it varies on how well versed is that physician in antimicrobial stewardship and how strongly do they believe in the antimicrobial stewardship, and it's a personal preference, sadly, in many cases. That said, when you start treating patients with a broad spectrum antibiotics, we're now digressing on a different topic, but usually you have to have some understanding of that there is a cause. So your patient actually should be symptomatic when you start treating them with broad spectrum antibiotics. And this is when we start looking into these cases that present to us. They do have symptoms, but we don't know what is the cause. So they're presenting with symptoms. So they have fever, they have something going on, there is some suspicion of infectious process undergoing, and then they put them on a broad spectrum antibiotics. With broad spectrum antibiotic, the problem is that with using them, we are just breeding more into this the resistance and supporting more of the resistance of microorganisms. All right, so let's go back to our micro piece. So with a host, what is important for us to know, because we're dealing in this situation with humans, is a couple of things that do affect this relationship between the host and environment and the infectious agent. So virulence is the measure that the microbe has the ability to invade a host and make a host sick. So with the humans, we have some of the defense mechanisms. So we have our skin, we have mucous membranes that do create the barriers and protect us from the invasion of the pathogens. So when you get your infections, like group A strep, for example, is after you had some sort of an injury on your skin, and people usually garden, and that's when you see that group A strep is actually entering through those injuries. When the skin is intact, you can have anything living on our skin. It's not crawling into our body. That's our body's mechanism to keep us safe. So when you're looking into also the we have our host flora in our own organism or on our skin as well. It's really important to recognize that this flora also can be depleted when from there is different things that are attacking us from the environment, if I can explain it that way, and it does affect the balance of the flora. So usually you will find this with mucous membranes more so. I think I mentioned to you in previous slide about the candida, where you actually have that imbalance and something has affected the host in a negative way, so the microorganisms can actually take over. So usually you can look also at, so for example, I mentioned also C. diff, and those are some of these imbalances in the human host that do happen and lead to infection. And then also when you're looking at, it's an actually interesting factoid here that our skin actually does secrete some of the short chain fatty acids that are themselves actually lethal for many pathogens. So we do have different ways of defending our own body from being attacked by the microorganisms that are around us. Oh my gosh, this is so teeny tiny, I apologize. So basically what I have put down there is just a number of microorganisms that we are encountering in our daily life. So you, on the left-hand side, you have the lactobacilli, which is a beneficial microorganism, and then as you're moving towards the right-hand side where that angry face is, there is actually CPE and ESBL, and then there is in between a range of microorganisms that do affect us negatively. So not all microorganisms are bad, but not all microorganisms are good either. So even with the some that we would say they are somewhere in between and they could be harmful, it all depends on how we as hosts respond to them. So there is a couple other terms that I think you need to be comfortable with also for the purpose of our exam and the CIC exam, is the difference between the virulence and pathogenicity. And pathogenicity is actually ability of a species to cause a disease. So that's the ability of the microorganism to cause a disease, where the virulence, it actually refers to ability of a strain to cause a disease. So when you're looking into the factors that influence the virulence, you're looking into survival in environment. So is this microorganism that will have ability to survive for the extended period? So we are thinking, I immediately think of microorganisms that have ability to create spores. How effective are they in transmission? How good are they in evasion of the host defenses? So talking about our own defenses that we have as a part of our makeup, are they good actually avoiding all of that and finding different ways to survive? Are they actually resistant to antimicrobial agents, and are they one of those that produce toxins? So all of this affects the way we have a relationship with the microorganisms. So we're going to go back and take a look at the environment. So how does environment affect this relationship? So some of the microorganisms, as I said, they have ability to survive in the external environment. Some of them cannot. So you will find that some will create spores. Some do have a more of a lipid envelope that help them survive in unsafe environment. So when you're looking at some of the microorganisms that are successful in surviving, you can think about your hepatitis B, hepatitis C, HIV, cytomegalovirus. What is interesting about mycobacterium and tuberculosis, think about it, it's predominantly a respiratory virus, although it can live in other areas in the body. They actually have developed this lipid kind of a coat that they wear to prevent from drying. So when humans breed mycobacterium out of their system, they are able to survive due to this capability that they develop this lipid coat that protects them from drying in the air. And then you're looking also at Pseudomonas. They actually can survive in water for extended period of time, again, because they learn how to go into almost this hibernation state where they're not using up all their nutrients in the survival period. All right. So the common routes of transmission, there are mechanisms of entry and portal of entry, and then some of the examples. So you can read for yourself, but there is usually the first thing that comes to my mind that I think of is for direct contact is MRSA, and it does have entry is skin. Can also be in the lungs. MRSA can be found in the lungs as well, can also be found in the blood. But for most colonizations, when we are talking about MRSA, we are talking about the skin colonizations. So to highlight a couple of things here, I would say people usually ask me, oh, does it only live in the area that we swab? Because we tend to swab nasal passages and perineal area. It actually doesn't just live there. It's just the most yielding environment. So it's nice and warm, and it does have all the nutrients that we can have the highest yield when we are swabbing our patients. So then you're looking into your sexual intercourse, through genital entry, syphilis. So as you can see, there is different ways of how can we actually acquire these organisms. I kind of always think about the coolest way to get something is the cooling towers and also the misters in the grocery stores, and that's for Legionella species. Legionella likes to live in the water. It's a water-loving organism, and it does get dispersed through these spraying mechanisms that we use for air conditioning, or you will find also Legionella in the heating towers for the hot water. And I find for most of the facilities that would have a big problem with that would be long-term care facilities, retirement homes, where the water temperature gets reduced to prevent scalding, and then we actually enable Legionella species to grow in these towers. A couple of other things. My animal bite also would be your rabies, and then tick bite for Lyme disease, and and for bacterial pneumonia also is your respiratory disease. So it's just a different way of looking into these portals of entries and what kind of disease they're causing. So those are your common ones. Okay, so I'm going to try this. I'm not going to play any video. I'm just going to narrate for you. Let's see if I can do this. Okay. Okay, so I'm gonna try and share my screen, and I'll try not to narrate. I'll narrate for you. You guys see the chain of transmission on your screen? Yes. Okay, so I'm going to talk. I'm not going to play anything for you. I'm going to show you how does this work. So on your pink side, you have the contact transmissions and the microorganisms that are most known to be transmitted through contact transmission. The green ones are the droplet transmissions, and the microorganisms that we are aware are transmitted through that route, and then airborne. So let's pick one. Anyone who gets first to pick one of the microorganisms, I'm going to take you on a journey through the chain of transmission. Anybody? Flu. All right. So let's take flu.
Infectious agent. Staphylococcus.
All right. So we picked flu. Kyle was first to get there. So then you can see that there is infectious agent, reservoir, portal of exit, mode of transmission, portal of entry. So for us, it's important to be able to break this chain of transmission. But let's take a look at what is infectious agent. So you see that influenza is caused by the influenza virus. So we know that, right? What's the reservoir? It's always humans. However, we also know that there is birds and other mammals. Most common ones that we come across that do affect humans are the pigs, actually. Okay. The portal of entry. How does this influenza virus enter our body? It is through our mucous membranes. It is nose, mouth, and eyes. What's the mode of transmission? So we kind of already know because we picked it. It's a droplet contact usually. So you can read about in the box. It's quite a lot to read there, but it's, you guys know how the influenza is transmitted. So somebody is sneezing or coughing or talking, as we know people do talk mostly. So sometimes those little particles will end up in our faces and then enter through the portals of entry. And then you can see here that the portals of entry are, again, your eyes, nose, and mouth, just like the portal of exit is. It's not in all cases that they're matching, but mostly they do match. And then who is the susceptible host? So I kind of like to ask these questions. Who is the susceptible host? And I always get surprised when I'm actually asking the frontline staff that they're not saying that for influenza, everybody has a potential to acquire the influenza. So people who are not vaccinated, even in some instances where you are vaccinated, we are still at risk because the vaccines are not always 100% working in terms of coverage. And then, so let's break the chain. So how do we break the chain? So you can either break it through any of these. So if you're thinking about the mode of transmission in terms of, there we go, now you can see that. How do we break that chain is by cleaning and disinfecting the surfaces, shared equipment, performing hand hygiene, also making sure people are wearing proper PPE and adhering to proper donning and doffing practices. So if you're breaking a portal of entry, again, for the healthcare workers is to wear PPE, but also to perform hand hygiene when in contact with contaminated surfaces or contaminated environment, or if they're performing, of course, the aseptic procedure. So I would just refer back to the 4 moments of hand hygiene in this instance. So if you're looking into breaking the chain of susceptible host, it is turning around mostly about that education piece and how do we educate who are people at risk. So talking about the hand hygiene again, talking about immunization, and also the respiratory etiquette. Okay. So if you're looking into breaking your chain at the link of the infectious agent, so it's important for us, again, to isolate that infectious agent and prevent it from entering the portals of entry. So how do we do that? So we have to make sure that, again, we are sticking to the additional precautions. In this case would be droplet contact precautions, and sticking to risk assessment. So we will cover that next week, more about the risk assessment and additional precautions in addition to the routine practices. But this is another important piece to educate everybody to know when to start actually critical thinking and being able to make judgments for themselves in addition to droplet precautions. So before we get to that point that somebody is isolated, it is critical that we use that critical thinking piece and we are able to recognize before the patient is actually on isolation that there is something that could put us at risk. All right. So then if we are trying to break the chain at the reservoir link, so this comes in place now, finally, that isolation piece, isolating in a private room whenever possible. That's not always the case. So sometimes we do have to cohort, and we will be talking about that a little bit more. But again, education, cleaning, limitation of the visitors, that always helps when we are dealing with outbreaks. It becomes a little bit more evident and we stick to those rules a little bit more during the outbreaks. But during the regular flu season, these visitors still do come in and do their thing with the families, and it's not necessarily the best thing. However, it's a fine balance between the infection control and the patient experience as well. So a couple of things would be, if you're looking into your portal of exit, educate again clients. So you will see there is a lot of repetition in terms of education, hand hygiene, and PPE. It's just how and where do you insert these, okay? So I am gonna stop sharing the screen, hopefully. Okay, let's see. Did you guys like the chain of transmission? Did that make sense? Yes, okay. So I tried to upload this chain of transmission. Maybe Leonard can help me later. I could not upload this for you guys in the shared documents. So I don't know how to do this. This is definitely beyond my skill set. I know everybody knows where my skill set is now. All right, so we are going to talk now about the immune system and what kind of so this is important. I'm just going to highlight this to focus and pay attention. It's these two different immune systems that we as hosts do have, and this is also something that perfect, Leonard, you're the best. So we have innate immunity, which if you actually think about the word innate, it tells you that this is something that we are born with, because innate means baby, and this is the immunity that we are born with. That's our first defense. It is immediate and it's quite crude in a process how the body responds to it. And then you have your adaptive immunity. So that's usually specific immunity that we develop over the years. Either it's due to exposures, through being sick, or getting could be also through vaccines. So for the purpose of writing your CAC exam, please get comfortable recognizing these two immunities and how they work and what they are. So as I said, our innate immunity is our first line of defense against the pathogen. So if you're thinking about innate immunity, think about injuries that happen. So if you had a scratch, or you have a puncture in your skin, and that first response that is non-specific immune system, that's our body trying to extract and push out those foreign bodies. It could also be a microorganism that that our body will be defending us from, but this is one of the first and most visible is actually when you see the injury and you see that redness and a little bit of heat. So usually that's how you can see actually your immunity in action. So I'm just going to highlight this again. Your innate immunity is going to be on your CSE exam, and recognizing the two and distinguishing one from the other rather. So when you look into your innate immune response, it's broken down into inflammation, and that's your first response. Usually shows up with redness, swelling, pain, and warmth. So if you think about again that injury, so say you were gardening and something has broken your skin, and now your body is trying to push out that little foreign object. So you will see the redness around the area of entry. Sometimes you will see swelling. It does hurt a bit, and it can be warm around that area where the injury has happened. So the goal is actually to prevent infection from setting in. And that is when you get your body mobilizes your T and B lymphocytes. So get to know your T and B lymphocytes and get to know how does this mechanism work. So I do have some videos. I will upload those videos after the class that you can look at how these the immune system rather works. So then when you get into your adaptive immunity, it's your humoral or cellular immunity. So those are the two different mechanisms that your adaptive immunity responds to. So your humoral antibody-mediated immunity, it's actually working with the assistance of helper T cells and B cells. They work together to produce an antibody, and that's actually an antibody that is specific antigen neutralizing and causes lysis. So lysis is actually breakdown or phagocytosis. So cellular immunity occurs inside infected cells, and it's mediated by the T lymphocytes. Your helper T cells release cytokines, and they help activate the T cells. So as I said, there is a video I will be sending you, and I think it will help you significantly understand how all of this comes in play. So we get into bacteria. So remember that slide that I was showing you, the gram positives and gram negatives. They're broken down into cocci and bacilli. So we are going to get into that. Do you guys want a 5-minute break before we break into the bacteria? 5-minute break.
Cornelia, I have to say, I'm very sad that you took off your little kitty cat headband. It was actually so illuminating while you're talking.
It's actually poo.
I noticed it right away.
So Jagdeep, one day I was wearing these at work, and a lady walks by me and she meows at me. I thought, okay, well that's kind of odd that she meowed at me. And I forgot I had these guys on my
I've never heard of somebody meowing, but I would definitely compliment you on it. That's so funny.
But I forgot I had these guys on my head, right? So I thought, like, that's an odd greeting. Okay, never mind. So I carry on by, and one of the nurses on the unit, as I show up, she goes meow.
Oh,
So I'm
God.
thinking,
That's so funny.
what is going on? Why is everyone meowing at me?
Did you forget you're wearing the headband? You forgot.
Oh my gosh, I completely forgot about it, right? So I'm just walking around happily with my little headband after I did an in-service. Yeah.
All right, cool. So we're taking a 5-minute break then.
Yeah, I think let's give people some more time. Maybe someone needs to go to washroom.
Okay, thanks. See you in 5.
And this, Corinne, and I didn't even drink anything, so you can imagine how would that be if I actually had some wine now. It's okay. I think people should enjoy the break with us too, whoever follows to watch. Because if I stop recording, I forget to start it up. So again, it's important that we have some laugh in a day. All right, so are you guys back and ready to continue with hopefully less hiccups with my presentation? All right. Yes. Oh, hi, Isha. How are you? I was wondering where is Marina? I haven't seen her being much active on this chat today. All right, so let's move to our gram-positive cocci. There she is. So they're broken down into staphylococci, streptococci, and enterococci species. You will probably in your day-to-day work see a lot of staph and a lot of enterococci, and I hope you don't get a lot of streptococci, but we'll get there too. So we'll prepare you for all of that. So the Staph aureus actually is, let me show you. Sorry. So the little guy, you can see even on the picture down on the screen, it does have this protective shield. It's some sort of a cape that they do have. But what is important for you to know is just a couple of things, that it is Staphylococcus can cause foodborne illnesses, and usually you will find these quick reactions to food when after somebody has eaten a food that is contaminated with Staph aureus. It's really quick turnaround, and it's a lot of vomiting, and it's usually in relation to poor personal hygiene of the food handlers. So they tend to put their fingers up their nostrils or sneeze in your food and things do go wrong, or they don't clean their hands before they handle. I know no one wants to hear about this now. Probably some people are munching on something, so I'll just skip on this. It is causing the beta hemolysis on your blood agar. So I put a picture for you guys to see. So when you look on the blood agar, rather, where the staph is growing, it is eating up the blood. So you will find that is a clear line around it. So it is a coagulase negative. So some of them are, as I said, it is staph epidermidis, which is of course living on the skin, and the other one that is lagdunensis, it's not actually hemolytic. So it's not all of the staph will cause the beta hemolysis on your blood agar. But I do like these little guys to show to people how they do have their little protective shield when they're MRSA versus Staph aureus. So when you're looking into the diseases that Staph aureus, that Staphylococci rather can inflict, so in regular population, as I mentioned earlier, you will find that about 60% do carry Staph aureus on skin, and that's usually just a colonization. So you will find that some people are intermittently positive. So it will be, you would swab them today and swab them after a certain period of time, and they will turn negative. And then they have this ability of self-colonizing or getting recolonized through exposures. About 20% are persistently colonized, and some of these poor souls are going to be coming back into your facilities, and every time you swab them, they're positive for MRSA. And as I said before, we do collect swabs for MRSA. Usually for colonizations, we are looking into the nasal swabs and perineum. However, if there is a suspicion that the wound could be also infected or colonized, so that's another thing that you probably need to know, is that wounds also can be colonized and not just infected. So it does require a physician to take a look at that particular wound, or a wound nurse to tell you whether that's an infection or colonization, and you would be collecting those specimens. So in our week 6, unit 6, we will be talking about the collection of specimens and how does one acquire these swabs that we need for testing. So Staph aureus, as you now are drawing the conclusion, it could be also a pathogenic organism. So it does live on our skin. When they're just happily living on our skin, they're causing no harm. That's called colonization. However, once they enter sterile sites, they causing infection, and that's when they're pathogenic. So they do, as I said, they cause bloodstream infection. They also can be found in the lungs. Usually you will find them in ICU in ventilated patients, that the MRSA or the Staph aureus actually enters the lung tissue, but you will also find it in GI tract as a part of the foodborne illnesses, but also can be entered into the bones and joints, usually through surgical procedures. Okay, so there's some pretty pictures for you to take a look at in case you didn't. Burnett, she's like, I'm getting dazed and confused with overwhelmed with this information. I need something to keep me up. So I just put a picture here of a graph for you, that how the MRSA actually is doing since 2000 till 2018. You will see that we actually have an increase in MRSA bacteremias in particular. This is so bacteremias are actually reportable, and we are mandated to report them to Quality Ontario, and that's used usually for benchmarking when you're looking to see how are you performing in comparison to the other facilities. You can access the Quality Ontario website and pick the neighboring hospitals for the bacteremias. Couple other things to think about is with regards to the collection of this information is it's the Quality Ontario and bacteremias are reported only by the hospital settings. You will not find it for the long-term care facilities or retirement homes. So as I said, you will find that if you're looking on this particular graph, that we are on a somewhat uptick there with the bacteremias. You will see also that's a very similar situation with the VRE as well. And there is also a bit of information for you to know that we do not report the colonizations to Quality Ontario. However, when we do experience outbreaks in our facilities, that is reported to the public health unit. And that's a different piece of different database. So the staph is actually coagulase negative, and then when you are looking into your coagulase negative infections, there is about 40 different, over 40 rather, different organisms that are part of Staphylococci family. So we already have addressed some of them. You can read them for yourself: Staph epidermidis, Hemolyticus, Saprophyticus, Schleiferi, and so on and so forth. They're mostly living on our skin. They're not causing any harm as per usual. However, they also can be opportunistic pathogens if they're introduced into the sterile sites. Usually you will find that in some of these cases, usually you will find the infants and the children in the NICUs that are affected by the coagulase negative staphylococcal infections. I think it's important also to bring to your attention that we do find them entering sterile sites through surgeries. I mentioned that before as well, but also indwelling devices. Back to how it stuff ends up in a lung. Yes, we should use more maggot therapy, Leonard. That's what we should be looking at. It's not really commonly used, but it does happen sometimes. I don't know if anyone has seen it as a therapeutic measure, but it is used sometimes. All right, so your Streptococcus species, I will actually tell you a little bit of a story here. So it is most common is the group A Streptococcus that you will be dealing with. So please do study about the group A strep infections. It is known to cause bloodstream infections and necrotizing fasciitis. Group B Streptococcus is predominantly linked with the pregnant mothers testing and infections in the newborns. So it is a gram-positive cocci. So here is the thing, how usually they would ask you a question that if you get preliminary test, they would tell you, so you got a gram-positive cocci, and it is presenting in certain fashion from the symptomatology perspective. So on your test, they may give you a list of organisms, and you have to pick the best fit. So usually they will give you of course the correct answer, and then there's going to be one that could be something that you would be thinking in terms of presentation perhaps, and then usually they will give you something that is completely off and has nothing to do with the presentation, and neither it's a gram-positive cocci. All right, so these are the streptococci that do affect humans. So they're broken down by the serogroups. So the Streptococcus pyogenes, that's your group A. It does affect humans. The reservoir is humans, and most common disease is actually the pharyngitis. That said, it will cause also sepsis. It will cause necrotizing fasciitis, and then your strep group B, as I said, that's for your neonatal meningitis and sepsis. So do know both for your CIC exam. I have never come across the rest of them being listed on the CIC exam, but this is the one. These are the two that you want to be quite comfortable knowing what's the host, who is the population at risk, what are the symptoms, what is the incubation periods, and so on and so forth. Okay. All right. So your group A strep, as you can see, there is over 240 distinct serotypes and genotypes of a group A strep. What is most common and least harmful is that pharyngitis. It does cause also a skin infection or impetigo. It does cause scarlet fever. It does cause the puerperal fever, if you remember from Semmelweis. That was one of his biggest battles that he fought. It does cause also septicemia, erysipeal, pneumonia, and neck fash. In your work as an infection control practitioner, I will tell you, you're probably going to come across some of that neck fash and some of the sepsis. In my work, I actually had been dealing with a group A strep outbreak in maternity ward. And what has happened is we had one mom that had a baby with the midwife program, and she went back home after she had baby. Everyone was fine. And she presented the following day on one of the satellite sites of our hospital with fever, and started having a bit of a fatigue and not feeling so grand, and they sent her immediately down to the main site. We have admitted her into ICU and collected the blood specimens. 2 blood cultures are necessary to test, and she came back positive for group A strep in blood.
You know what? When you're talking about that, Cornelia, I worked in labor and delivery, and it was a very sad time. Like, I was getting orientated, and that's when, like, you know, our leadership decided that they really need to give us education on this, because there was a mother who gave birth and her fever was up, but nobody had gotten blood cultures or done anything, and she actually died. And that nurse is forever, like, upset about it. She's, like, gone with it. And she would literally, anytime there would be anything over 37.8, she would be asking everybody, what do I do? What do I do? What do I do? Like, she lost her self-confidence. And this is a nurse that I knew that worked for 25 years.
Yes. So I will tell you what happened with our case, and I will share this because has been quite covered in the media for anyone who has been watching. So it was back it was actually during the winter months, and so that mom and baby were fine. And then about 10 days later, we had another mom. She was that was her 7th baby, and there were some language barriers and whatnot. So she was not really having a good communication explaining how she was feeling, and at some point she actually has spiked a fever. And to your point, we usually start looking at fevers commonly across the board as 38 and above, and it also usually takes 2 temperatures. And what is really unique for your maternity population is that they do tend to spike fever during labor. So it's often
actually, Cornelia, since you're talking about that, I actually have an OB that I know who will do a vaginal exam, and she feels the temperature, and she knows that this person is about to have a temperature. And it's funny because it happened to me 3 times. And so literally, like, she's like, this person's gonna have temperature. We're gonna have to have a C-section. Like, we're talking about this. And then it turns out she actually ends up getting a temperature, and I'm giving them Tylenol in between, and then trying to see, like, the fetal heart rate goes up, and then it's C-section. So it's funny, though, like, she has this magnetic thing.
So they do have their own ways of dealing with things, and not necessarily that is all related to the group A strep, right? So I think that's where it becomes really challenging to navigate through the murky waters of what is a true fever and what is the fever that is leading through the delivery process, right? So I'll just try to wrap it up so we can move along. So what happened is, so this one mom ended up having blood positive for group A strep, and then later on the following day, I believe, or it was 2 days after, we had another mom with that had delivered. She was a younger individual, had her baby, and she was not having really the typical symptoms at the beginning either. But what was really interesting for her, she was just feeling tired and asked for privacy. And so there was a little bit of a challenge keeping, I guess, a close eye on her because they closed the doors and so on and so forth. But what was really interesting is that through the investigation, we have found our moms having the group A strep. So we had to test further to identify whether they were linked and if there is a same genome. To the entire facility, we have swabbed close to 400 staff in search for the group A strep among our staff to identify if we actually had somebody that was colonized and not symptomatic working. So it was quite a lot of work for us. But what is, to your point, that you're saying is with the fevers, so we dropped down the threshold for these fevers. So we weren't waiting anymore for the fever to actually hit the 38 when we started screening our moms. So anyone, and including these what they're deemed as laboring fevers, we were collecting blood on all of our moms, 2 cultures, and making sure that we are not having any of these incidents happening. So it is quite heart-wrenching for the frontline staff when things like this happen, because it's in labor and delivery, it's all happy, right? It's all good news. Everyone has always good experience on the unit. So there's new life comes, and you're looking at the situations that are always so joyful. When things turn in a different direction, it does affect our frontline staff, and they do feel quite depressed about the events. And they, just like your example, they do blame themselves, thinking that they have done something wrong. But with group A strep, what is really important to know that individuals actually can self-colonize and cause their own infections. So sometimes you have the chronic carriers of group A strep that are carrying it in their throat. They're not having any symptoms. However, that group A strep resides there. And then it could be through the labor and delivery that, when you are yelling at the mom to push, that some of those droplets do exit and cause these infections. So something to be aware of. So when you're looking into your group A strep, always think about your healthcare workers, and that they also could be the carriers of group A strep. So when you're screening, if you end up, and I hope you never end up doing this, when you're screening your healthcare providers, you're always collecting a throat swab. For females, it's a vaginal swab or a rectal swab to identify if you have some of those asymptomatic carriers. So for your group B strep, so this is more common. I think this is what most people are common and comfortable in the labor and delivery. So we do screen mothers for group B strep before delivery, because of that vertical transmission during the labor and during the delivery, that it can actually cause infection in the neonates, and it's usually ends up in neonatal meningitis. So we do screen them, and then what you need to know for this, that incubation period is actually shorter than 7 days, and that about 15 to 35% of women are also colonized. So keep that in mind, park that stat in the back of your mind, because that also may show up on some of your exams. So then you get into your strep pneumonia. That's another one that I find it's quite common. You will find that fairly frequently on your results coming through. It is a gram-positive organism. It does actually cause an alpha hemolytic hemolysis rather. So what alpha hemolysis looks like on the screen is more kind of greenish gold color on the blood agar. It's not that clear, but your beta hemolysis does to the blood agar. So you will find these greenish stains on your blood agar. So that's what that is. And it is causing it has about probably over 90 different species of pneumococcal serotypes. So that's neither here nor there, and that's a statistic that you really will be asked ever in your life about, but it's good to know. And then you also should know that the children are common carriers. Depending on where these children are coming from, depending on the socioeconomic status of the country, you will find that as the socioeconomic status of the country is dropping, the children are actually more affected by the strep pneumo. And it does cause pneumonias, cause meningitis. It can cause also bacteremias and sepsis. And there is a vaccine that is available, and it is recommended for the patients with splenectomy. So I'm going to repeat this. There is a vaccine that is available, and it is recommended for patients with splenectomy. Incubation period is 1 to 3 days. Burnett, how's that vodka going? All right. So the next microorganism that we are going to touch is actually Enterococcus species, and there is about 18 of them living in that big family of Enterococcus. Most common ones that you will be seeing is Enterococcus faecalis and Enterococcus faecium, and it is a host, and it's rather commonly found in a host, in a GI tract of a host. It is a normal flora of our GI tract. The problem arises when it transports from the GI tract into your urinary tract or blood. It can cause also abscess. It is highly antibiotic resistant, so you know it as a VRE when it is antibiotic resistant, and it does require dual therapy for different for the infections. There is no clear incubation period. We don't really know what is incubation period for this particular organism, and you will also recognize that across Ontario, we had a lot of hospitals at some point that dropped off and stopped isolating for VRE. And shortly thereafter, Public Health Ontario has conducted a surveillance across, and what we actually have found out is that there was a significant increase of VRE infections, bloodstream infections. And since that stat has been recorded, Public Health Ontario is recommending contact precautions for VRE. However, as I said, these practices do vary. All right. So the next topic is your bacillocentrusis, and it is a aerobic gram-positive organism, and it is spore forming. So if you find it in an environment that is not the most hospitable environment, it will create a spore to survive. It is a non-motile rod in shape. So it does cause skin anthrax. It can also cause a lung anthrax or gastrointestinal anthrax. And from your CIC perspective, it's probably one of the most frequently used as a bioterrorism tool. So the incubation period can be as long as 43 days. However, for the skin or the gastroanthrax, it actually can be as short as 1 week. So as I said, it is usually, if you ever think about all the scares of whenever the envelope is sent to somebody, they usually say it is a powder form. So, and it's really a good microorganism to do so. That sounds awful, but it is the spores are highly stable, so you can actually transport it easily, and for that purpose alone is really good as a biological weapon. So do remember that it is used as a biological weapon. So when it comes to transmission, there is rarely documented human-to-human transmission. However, if you ever come across a patient that does have bacillus anthracis, it is contact precautions for contact with the skin lesions. And I hope you never come across that. So there is Bacillus cereus, and that's another rod-shaped organism. It is a gram-positive organism. So I have a rice image of rice there, and the only reason I have it there is because it's commonly linked with foodborne illnesses, and it does cause poisoning. Actually, it does cause diarrhea and emesis. So it does have the ability to produce toxins. And if you look at the incubation period, so do make a note that this is probably the shortest incubation period. You may be or may not be asked about this incubation period, but it's about half an hour after you have eaten that fancy rice with Bacillus cereus that you actually can become symptomatic for the vomiting presentation, and can take longer, of course, for the real presentation. We do not initiate additional precautions once we know that the cause is Bacillus cereus. Usually the patient presents with diarrhea and vomiting, and we would initiate additional precautions. But once you find out the causative agent, and if it is Bacillus anthracis, you can discontinue those precautions. All right, so Corynebacterium diphtheria. So I kind of like the look of the little microorganism. However, it's an awful disease that does create membranes, pseudomembranes in one's throat, which prevents you from breathing. So that's one of the typical symptoms of the diphtheria. And it is a non-motile organism, so I don't think anyone's going to ask you about this particular organism with regards to the motility. However, do know that it does have this ability to create membranes, and sometimes I heard physicians say you actually can penetrate that membrane with a finger. Not that I would ever dare to do that, but I've heard people talk about the ability to actually penetrate this pseudomembrane that creates in throat. And sometimes you can also find it in people's noses as well. It does cause also ulcers on the skin. But what is important is that there is a vaccine that is available for use. And if you come across a patient that does have diphtheria, it is droplet contact precautions, and the incubation period is about 2 to 5 days. What is important to know is that even post-recovery, one can shed this particular organism up to 6 weeks. All right, so I don't know if you guys remember years back when we had listeria issue with maple leaf, but that's one of the, I'm trying to make you remember. If I'm sure some of you were way too young to remember that, but some people that are my age may actually have been lucky to work with people that actually had dealings with listeria. So it is a facultative anaerobe, and I think for you it's important to know that will survive in the refrigerators. So it is usually related to food that is processed in the manufacturing, such as hot dogs or deli cuts or unpasteurized dairy, and also has been found in ice cream. But a particular event with the maple leaf was in relation to the deli cuts and their smoked meats. It is a foodborne illness. However, once one acquires listeria and it's actually diagnosed with listeriosis, you can, the mortality rate is about 20%. So it's high fatality rate in case one acquires this disease. It does affect negatively the pregnant population, so usually causes a stillbirth, and that's why we are not recommending pregnant women to eat any of the soft unpasteurized cheeses or cold cuts, hot dogs, and so on and so forth. So something to think about. If you have any friends that are expecting, just remind them. And then with the incubation period actually varies, so this is really interesting to know that with pregnancy-associated cases, that incubation period can be between 2 to 4 weeks, where in population that is not pregnant, the incubation is about 1 to 14 days. So for pregnant women, incubation period can be longer, so it should not be excluded that potential exposures if you're dealing with any sort of community outbreaks. So back to some of that symptomatology in cases that are contracted listeria, you can see patients present with meningitis, encephalitis, endocarditis. It is a self-limiting disease, so there is no treatment for it. And important to mention how many people were affected by the maple leaf outbreak here is because of the distribution, how widely distributed was the product and still is. It's across Canada. There is 56 confirmed cases and 21 fatalities. In Ontario, there was 41 cases and 16 deaths. So Martina, no more deli cuts for you, okay? We always want what we can't have. That's why Burnett was talking about vodka. Martina. All right, so we are going to get into a little bit of a different discussion here. So we are going into your gram negative. So you're one of the most common ones that you will be coming across, and you should be comfortable knowing about this particular organism for your test. It is a gram negative diplococci, and that's it's one of the organisms that has also 13 serogroups, and it's based, the serogroups are actually identified in the capsular type. It is aerobic, but it can survive in anaerobic conditions, which means that prefers aerobic environment, but has ability to survive again in anaerobic conditions. It does cause meningitis in children. And it does cause also sepsis. It tends to have a somewhat seasonal character. So you will usually find it that does closely follow influenza and influenza outbreaks. I don't really understand myself what leads to it, and I couldn't really find the reasoning for it. So if Martina maybe knows, or Anita, or whoever is that has maybe some insight, if you can share. I couldn't find any good reason why does this happen. It could be in relation to the closed environment during the winter months, that we are seeing people are spending more that time in a closed environment and close together. What is really important is that in a household contact, you will see that the infectivity rate is 500 to 800 times higher versus general population. So it is easily transmitted in those close contacts. In 2019, in Ontario, we have identified 36 cases. 32 were hospitalized. So you can see that it has a high hospitalization rate, and also ended up with 6 fatalities. In case you do see a case of meningitis coming into your facility, it is droplet contact precautions. Also good to know that there is a vaccine available. It is responding to antibiotics, and it's very sensitive to antibiotics, so those are good news. And then we also recommend the prophylactic measures to be put in place for healthcare providers that have provided close contact with respiratory secretions without PPE. So now we are getting into the Enterobacteriaceae, and it is a fairly large family that you will encounter whether you work in long-term care or retirement home, or in acute care setting. Those are fairly common to be found in your lab results. So they're either Enterobacter species, Escherichia, Klebsiella, Proteus mirabilis, Serratia. Serratia has a big impact on NICU population. You don't want to ever hear that there is Serratia identified in the NICU population in your facility. No one wants that. And then there is Providencia and Salmonella species. All right. So with regards to Enterobacteriaceae, they are potential pathogens. They're facultative anaerobes, and they do tend to be enterogram-negative bacilli. So you will find them in predominantly in the GI tract. Some of them will cause respiratory infections. Often they're found as causing infections of GI tract as well, and UTI, and then they can also cause some pneumonias, depending on what are we dealing with. So E. coli, so this is, I really want to stress for you guys, this is a slide you want to probably mark and be able to come back to, because it is giving you some important information. So when you're looking into E. coli, there is different types of E. coli. So you have the one that produces the Shiga toxin, or it's also shortened as STEC. People will sometimes say STAC or EPAC. So it depends on how people like to pronounce their abbreviations. So you have enteropathogenic, enterotoxigenic, enteroinvasive, and enteroaggregative.
Oh, here she's back. Oh, I think she's frozen again.
All right. So Rosemary, can you hear me?
Oh, I can hear you now. Yes.
All right. So let's move along with this slide. So I'm just going to say that please make sure you know information from this slide for the purpose of examination. So when you're looking into your Shiga toxin producing E. coli, it does cause hemorrhagic colitis. And in terms of hemorrhagic colitis, that means that is creating or producing usually bloody, but it could be also a non-bloody diarrhea. However, with Shiga toxin, you will more often find that bloody stool. So when someone presents to your facility and say they have bloody stool, usually it means that they have E. coli. It could be other things, but that's one of the things to think about. So when you're looking into your enteropathogenic E. coli, usually they present with these watery diarrheas, and they are usually acute in presentation, but they also can have some of the chronic presentation. It could be, it's usually related to some of some of the socioeconomic status and also the stool management in public. It is the next one that we are going to highlight here is enterotoxigenic E. coli, and it's also known as traveler's diarrhea. So it does present with watery stools, and it is the producing the enterotoxin. So again, that's toxin itself is heat stable, but it's important also that when someone presents with watery diarrheas, that you also should think whether they traveled, where did they go, have they been to endemic areas. So with enteroinvasive E. coli, there is the presentation is again with could be your bloody diarrheas, but it's also sometimes you will find that mucousy discharge with the stool. And then your enteroaggregative E. coli, it causes diarrhea. It affects across the board, all ages. It doesn't really have a pick, and it's usually watery, but it can be a bloody diarrhea in presentation. All right, so make sure you guys know your E. coli's with different presentations. I would say probably the ones that do circulate most often in question would be your traveler's diarrhea and your hemorrhagic colitis. All right, so I don't know. Now I'm taking you even further back in history. So the ones that do remember Maple Leaf, I don't know if you will remember the Walkerton event, but in Ontario, there is a little town that has a population of 4,800 people, and they all fell, well, not all of them, half of them, pretty much 2,300 actually fell ill due to exposure to water from a well. So the town itself had about 5 wells that were pulling the water from, and one of these wells was actually on near a farmland, and the runoff after the rainfall has brought down the manure, cow manure, into this well and contaminated the water. And it is the Shiga toxin producing E. coli identified. It's O157H7. So when you are looking at reading these papers, you will find that they all have used the E. coli O157H7 short form for this particular organism. Even in the newspapers, everyone was quite comfortable using this this name for this particular organism. So what actually has happened, it started off that people started presenting with non and bloody diarrheas, and they had the colitis, and after 2 to 3 days, it was across the town spreading. So it's been quite followed in the media. It's been the reports were written by the government, what went wrong, what can be done. And there's a lot of recommendations and a lot of paper and regulations that came after this particular event. So when you're looking into this, the actual outbreak, it was the way they were running the outbreak, it was not all the patients were actually tested. So about 174 cases were presumptive positive and 167 were confirmed positive. 65 of them have been hospitalized, and 27 developed the hemolytic uremic syndrome, and 7 of them actually have passed away. It was a really interesting case, and it's always interesting for public health folks to go back and review. We like to look at it from the forensic perspective, how it actually happened and what could have been done to prevent it. But a couple of things that came out of it is the recommendations in terms of the well management, water treatment plants, and also protecting the water sources. So the next organism we are going to cover here is Salmonella. And Salmonella is another organism that you will find quite frequently causing foodborne illnesses. It does actually fall under the family of Enterobacteriaceae. And most commonly, it's linked with chicken, any sort of really animals that do lay eggs, including turtles and snakes. So you will find them often in relation to petting zoos, and also not just your typical chicken, not when it's chicken, of course, when it's raw, but so it's also the eggs. So Salmonella is actually found in the gut of the chicken. So if you know anything about the anatomy of chicken, it's kind of like everything comes out the same way: the feces, the urine, and the egg. It's they have this body part, I would say, that's called cloaca, and everything is coming out. So when the egg is actually forming, the shell is soft, because their feces and everything is traveling through the same channel. It does actually have ability to get the salmonella into the egg. In Canada, we consider all our eggs to be at risk. So we are always saying all eggs have to be fully cooked, and all chicken has to be fully cooked and prepared to prevent salmonellosis from happening. So So Sharon, I'm so sorry that you feel that way, but there is more coming your way. So I suggest you eat right now what you can, and we'll take a look how we're shaping towards the end of the course. So with Salmonella enterica, there is six different subspecies, and as I said, it is most commonly related to enteric diseases. It is an oral fecal transmission. What that means is that the host actually has to ingest the fecal contaminated matter in order to get Salmonella in their organism. So usually that can be also through cross-contamination if you are preparing and cutting raw chicken and using same cutting boards to cut your lettuce or bread, and that's how you get that in. Often during some of the baking processes, some recipes, old-fashioned recipes, will call for raw eggs. Avoid at all cost. There is ways to incorporate your pasteurized egg. After all, Pasteur did not fight out there for nothing to tell us that we can actually grow bacteria in non-pasteurized environment. Leonard, we are not going to tell them anything about the coating that we are putting on the fruits and vegetables to have the nice shiny appearance, like apples, right? We are not going to share that with them. And there he goes. Okay. So one of the historic events that everyone pretty much knows, I'm sure everyone has either been called at some point Typhoid Mary, or at least felt that way. I know I do feel some days like Typhoid Mary, but for completely different reasons. Luckily, not like this poor soul. But there's this lady, Mary Mallon, and I actually have uploaded a little bit of her story on our page. You can read about this unfortunate soul. But she was one of these asymptomatic carriers, and she carried Salmonella in the gallbladder. And what actually happened with her, she was employed by some snooty New Yorker over the summer to cook for the family. And she has caused some serious outbreak in this particular household. So from the August 2nd to September 6th, 6 of the 11 people presented with typhoid fever. And all along, she was fine. She was not getting sick. She was just being herself, prepping food and doing her own thing, while the rest of the people were falling ill. In about 3 months, about 25 people actually have been diagnosed with typhoid fever, and including some of the physicians and nursing staff, 2 of which sadly have died.
Sorry, Cornelia, for typhoid, what is the actual source?
Salmonella typhi.
Oh, okay. Thank you.
Yeah, no problem. So it is Salmonella, and that's what is the problem. So with Salmonella typhi, you can find that people are not presenting with symptoms, and they can be shedding, and they can be lifelong shedders. So if you read about this particular poor lady, she's been chased around and around to be confined, and she was quite stubborn and changed her name to continue living her life. And for something happening so harsh, the way she was handled, I think something for us as healthcare providers also to think about in terms of the management of some of our patients and how we putting them in quarantine, to me resonated in that fashion, how sad it was that she was completely tortured. So with Shigella is another organism that you will find. It is not something that I have come across a lot in my work. It doesn't mean that some of you will not. But what is interesting about this particular organism is that it's most commonly found actually in ducks and geese. So in the communities that do tend to eat more of a duck or geese eggs, you will find more frequently these Shigella outbreaks. I want to say about 2 years, I apologize, 2 years ago or something like that, there was an outbreak going on in York region, and they never really nailed down the source. So whether people didn't actually know that they were eating any of the products that contained contaminated um, food. It's a possibility that, but there was nothing was common except with a couple of people. They were actually gathering in parks, and some of them had exposure, or common link, I would say, not exposure necessarily, was a water park. But then again, not really a mode of transmission, that's just the presence at the water park, unless people actually ingested some of that potentially contaminated water, because keep in mind it is geese and ducks, so it could be that something came from the geese at the beach or something like that. But things that we never find out, sadly. It is always fecal oral transmission. So again, the same thing. One needs to ingest food that is contaminated with feces in order to get the disease. It is a fairly low inoculum that's required. You're looking at few microorganisms to cause the disease. It's also presenting with this kind of slimy, bloody diarrheas. It can be up to surviving actually up to 30 days in water with Shigella. So again, that's why it likes that duck and geese, and where you're going to find them more than in the other egg hatching animals. Incubation period is fairly short, and we also do not isolate for shigellosis in healthcare facilities just because of the mode of transmission. Okay, so the next organism we are going to touch on is Pseudomonas. And with Pseudomonas, what is really important for you to know, it is a water-loving organism. So we'll find it very often in drains, often causes infections in patients in ICU who are on ventilators. It is an opportunistic organism. It does cause infections, mostly in immunocompromised hosts, but most common actually exposure is through the hot tubs, and you will find it that people will present with this redness on their skin, causes folliculitis. It is antibiotic resistant, sadly, and in the lab setting, if you ever are lucky enough that you can enter one of the labs, and have a chance to smell it, it does actually have, people say it's kind of grapey, but to me it smells like linden tree. So if you ever have a chance, and the linden trees are just about to start blooming, so I always think about Pseudomonas when the linden trees are blooming. Yeah, so you see, Anita says it smells like grapes. I think I may not have a fully working nose. I don't know. So couple of other organisms that I want to talk about are your Acinetobacter baumannii and Stenotrophonas maltophilia. I cannot speak anymore. They do colonize your gastrointestinal tract, and sometimes you will find them in a respiratory tract. Again, water-loving, finding them in drains, opportunistic, and antibiotic resistant. You know, Anita, what one of my professors did to us when we were in school, so she gave us to smell the Pseudomonas, and right after that, she gave us to smell E. coli.
So Cornelia, I might be jumping ahead of time. I'm just wondering, I know you talked a lot about with the CSE exam to know the incubation period, how long you're infectious, when you can take them off. I haven't had a chance to review anything, so I can't say anything for this week. But I mean, is there any point today or tomorrow that you're going to talk about what that is? Because I have a lot of info on what it is, but I don't see anything about how long you're infectious until.
Oh, so I will. So there are some of these diseases that are self-limiting and they really don't have a duration. As we are going through, I will highlight the ones that are unique and that you need to know. So as we are going through, you hear when I say, make sure you are comfortable with this, make sure you know. So I will be highlighting some of those important things for you. So Marina, E. coli smells like poo. So the Burkholderia cepacia, it's cause actually of cystic fibrosis in patients. So again, rather other way around, the cystic fibrosis patients can actually become colonized and they're at risk for pneumonias. I'm sorry, I'm trying to follow the chat and I can't follow my own train of thought anymore. It is highly antibiotic resistant. So this is an important piece for you to know, that is highly antibiotic resistant. Oh, I did not know that people smell things differently from different parts of the world. That's interesting. That's maybe why the Pseudomonas smells like lindens to me. So back to our Burkholderia cepacia. So in case of the cystic fibrosis patients that do get colonized or infected with it, they should be actually placed on contact precautions while they're in our care. So that's important to know and keep that in mind. It is a highly antibiotic resistant organism. So Legionella is another organism that is interesting and important from many perspectives, but it's going to be also mentioned on your CIC exam. So do know Legionella inside and out. It does cause actually two different diseases. It will cause Legionnaire's disease and also Pontiac fever. So what it is a water-loving organism. It does affect the respiratory system. It does multiply in water. And when does that happen? It takes place at the temperature that is between 25 degrees Celsius to 42 degrees Celsius. Incubation period for this particular disease is 2 to 10 days. However, Pontiac disease incubation period is about 1 to 2 days. It is a very similar disease. The only difference is that Legionnaire's affects the elderly population and Pontiac's disease affects younger population. It lives in the water, and it usually you want the water that is being used for misters in your food, in your grocery stores, for spraying fruits and vegetables. That's where you find that is frequently colonized with Legionella. So you want that water to be treated properly so you're not spraying any Legionella to your customers. In larger city dwellings, it is commonly linked to the cooling towers. So there was a big outbreak in Hamilton in recent years. There was an outbreak that was found for the spa, actually. There was a spa conference. And when I say spa, it's your hot tubs conference, where they had a different manufacturers came in and brought in these hot tubs to show, and they were having all the new developments shown about these hot tubs. The problem with that is that because the intent was only to show and not to use, the water wasn't treated and wasn't heated either. So through the jets that were aerosolizing the water, the attendees of the conference have ended up actually with either Pontiac or Legionella, Legionnaires, depending on their age. But there was outbreaks that were related to that. And then what is important to know from the IPAC perspective, there's no human-to-human transmission. You have to have that source that is spraying the water out. So it's something is causing the distribution. So it could be as I mentioned before, usually you will find in places such as long-term care facilities, retirement homes, they tend to reduce the water temperature to prevent scalding of the residents, and that actually permits for growth and multiplication of Legionella in this water. So usually when they open the water for the shower, that initial splash is actually what also is a problem with, even in the water tanks that are heated to the proper temperature. So we always advise to, when you're running the shower, while you're waiting for that hot water to come, just open the shower, don't step in. Not that anyone wants to be showered in a cool water or cold water, but don't get in immediately after you open that shower, because you could actually have something growing in those pipes as well. So there's no additional precautions. We are not isolating our patients that are diagnosed with Legionnaires. And then this is important. We send urine samples for testing. You can also send blood or sputum to look for the antibodies, but we send urine samples when we are testing for Legionnaires. This is important to know. All right, so then we are going to move into the Vibrio species. So Vibrio organisms are facultative anaerobes. They are motile. So this is important to know that they are motile. And if you look at this little picture, this little guy has a little tail. So that helps him move through the water. They do love salt water. So often you will find that they do affect the shellfish. You will find it in various shellfish that is served. If it's not properly cooked, you will find a lot of infections that actually do stem from it. Quite common. I know we have some people from BC. There is few outbreaks that have been on your end of the Canadian coast that were related to Vibrio cholera. They do have a toxin. Yes, they do have a flagellum. Yes, I like to call it a tail. So they do survive in salt water. Right. So Kyle, you're from BC, right? So do you remember any of the outbreaks in recent years that were caused by Vibrio cholera? Very good. And there is no human-to-human transmission. And again, very short incubation period. But it does cause some of the septicemia. Predominantly, it is a foodborne illness, again, but it's something to keep in mind. And if you ever want to watch a movie that speaks about Vibrios, it's the love at the time of cholera. So you can watch that. That's a lot of loving and a lot of diarrhea happening in that movie. So the next organism we are going to touch is Campylobacter jejuni, and it is, again, another foodborne illness causing microorganisms. They also can be drug resistant. it is relatively short incubation period, about 2 days. On occasion, you will see something longer as about 5 days. It does cause diarrhea in affected population. We do not isolate. The reservoir is usually chicken, poultry, unpasteurized milk, and untreated water. So if you want to, just something to show you. So if you look at the Vibrio, and they do have their little tail, or as Leonard likes to call it properly, flagellum, and then it says it is a motile organism, and so is this dude too. See, he has also a little flagellum, and it's also motile. I don't know, I assume it's a male microorganism. I don't know why. It's probably my language. So then we are gonna touch on some of the other relevant gram-negative bacilli for you. So Helicobacter pylori and Haemophilus influenza. Haemophilus influenza causes commonly pneumonia. It can also cause meningitis, where your Helicobacter pylori actually causes ulcers, and is commonly found in soil. So that's just something to add on. Now we're going to touch a little bit on anaerobes. So those are the organisms that live in absence of oxygen. And so they are also broken down into different categories. So you have obligate anaerobes. They cannot survive in presence of oxygen. And then you have some that are aerotolerant. So they can grow, but they survive best in the absence of oxygen, but they can survive it. And then you have your facultative anaerobes. So they will grow equally well. So they're either or. They're not too fussy about the oxygen, but they preferred their anaerobic environment. Where do you find them? You will find them in all mucous membranes. They can be found in skin. They do live in the gastrointestinal system. And when you're dealing with anaerobes, when you're looking into collection of your specimens, you're always thinking about the aspirate. So you're not taking your surface specimen. You have to go and get that specimen where no oxygen has made its presence. So some people will say they do have a specific smell. Again, it's something to definitely avoid, but they will say that also the infections that they cause are quite stinky. So when we are talking about these anaerobic organisms, it's important that we ensure that we provide them with a proper environment, not because we want to grow them, but because we need to test them. So when we are transporting them, we don't want to introduce any oxygen, so we have to make sure they're transported in special tubes. To culture them, you will usually wait for some time. It can be about 48 hours. In some instances, it can be even longer if you're looking for the subcultures. And usually when we start treating them, and that's your empirical treatment before we actually know what we're dealing with, we start with metronidazole. Who are the guys that are the most common anaerobic gram-positive bacilli? So know these guys, know the names and know what they cause. So you have your Clostridium tetani that causes tetanus, Clostridium perfringens, commonly known to cause gas gangrenes. And I will tell you that few of the questions on CIC that I had were about Clostridium perfringens. There is your Clostridium botulinum. It causes botulism, usually linked to canned foods. You will find it in foods that are canned and not properly processed, or when the foods were not properly cleaned and the spores actually have entered the can. So if you see ever a bulging can, that's a good indicator that could be actually growing some Clostridium botulinum in the can. And then again, probably the most known and most encountered in healthcare would be Clostridium difficile, which actually has now changed the name, and it's still shortened for C. diff, or you will find it as CDI and CDAD, which is Clostridium difficile disease, but it's always the C. diff causing disease rather. So as you see, I mentioned it is changed the name. It's called the Clostridioides difficile, and it makes it more difficult for me to pronounce all of these organisms. But let's stick with C. diff. So it's a spore-forming organism. It is an obligate anaerobic, and it is a gram-positive bacilli. So it does actually create and produce toxins. So this is important for the diagnostic piece. When you are looking into your patients, they will have often these colon that is affected by the toxins. So they're called megacolons. They're enlarged and affected by the toxin that is sloughing off the mucus of the colon. It is quite frequently found in the healthcare facilities. It does have a link to use of antibiotics. So you will find that patients who come into facilities come, say, with pneumonia, for example, and we treat them with antibiotics, or, for example, some of those cases that you guys mentioned, the urinary tract infections, or just a positive culture where we are persistently treating our patients with antibiotics. C. diff is one of those that is fairly resistant to antibiotics, and I mentioned before, it is not a good competitor. So it will overtake the gut once you kill off all the good flora in the gut, and then the C. diff takes over. What happens when you have a lot of C. diff in the gut? It actually attaches to the mucus and starts bringing all the water out of the body, and that's what you get, the very liquid frequent diarrheas. In some instances, you will have patients having up to 20 liquid diarrheas in a day. That's not uncommon. And it's also frequently linked to your healthcare associated outbreaks. What is interesting to know is that children under age of 1 are actually quite frequently colonized. There is some studies that are indicating that about 50% of kids can be colonized and actually have to be completely asymptomatic. So when you're dealing with kids and you find that they're coming in with C. diff, nothing to be alarmed, because they tend to be colonized with it. There is also talk about the adults being colonized, and that can be about 3 to 26%. That's quite a range there, but that's what the APIC book is actually reporting to us. Usually for treatment, you're using your metronidazole and vancomycin, and stool transplant, we already talked about that. And it's usually when you're doing your stool transplant, you also want to make sure that your patient is also followed with antibiotics as well. And it is always on contact precautions. You want to ensure that the sporocytals are used to clean, because it is a spore-forming organism and it has a tendency to survive for extended period. Another thing that's really important, it is coming as a part of fecal contamination, and feces as itself comes quite greasy. So it's important that you're using also good surfactants when you're cleaning surfaces to lift that greasy components of the stool that C. diff actually lives in. Sharon, how are you doing? Did you decide you're done with all the food for now? So next organism we are going to touch on is tuberculosis, and this is also going to be a big component of your CIC exam. So please make sure you are comfortable with this particular topic. It is caused by the Mycobacterium tuberculosis. It is an acid-fast bacilli. So this is important to know. It is not something that stains by gram. It is an acid-fast bacilli. You will see it under the microscope. It creates these neon colors or fluorescent colors, if you wish. And when you're running your test, you will actually see this that it comes. That's how they're reporting it. There you go, Sharon. You're gonna be a superstar to share some information with us with regards to TB. So when you are looking into the incubation, it is about 2 to 10 weeks post-exposure. And what we are looking at is the post-exposure is the TST test, which is your positive skin test. And I'm actually going to try and show you. This is the little tool that we are using to actually measure indurations. And so when we are looking for the skin tests, it is important to know that we are looking at indurations of 10 millimeters. That's about that. And induration is a swelling on the skin where the we actually have introduced the agent. So you will find this little welling almost on the skin in positive patients. That doesn't mean that they're infectious. That only means that they have been exposed to TB. When you're looking into TB, there is two different presentations of TB. So you have something that's called latent and active TB. So do make sure you're comfortable with both terms and knowing that just because someone's skin is testing positive for tuberculosis, it doesn't mean that they're actually infectious and actively spreading TB. So to cover that piece, latent TB is defined as an infection in a person who has tested positive for that skin test. There's no physical findings. So if you have, they're not going to be symptomatic, and their chest x-ray is clear. There is nothing on their chest x-ray, and therefore they cannot spread TB. It just means that they have had contact in the past, or they may have had an infection in the past. Sometimes people don't know they had it if it was a milder presentation, and usually with that first exposure, it is. With your active TB, your first signs are that abnormal chest x-ray, and typically described in the books, it's usually affecting your right upper lobe. Other important term for you to know is miliary TB. And that means that's disseminated extrapulmonary TB. That's when you get TB that is the body that's completely ridden with TB, and it's everywhere. So that's not to be confused when you have TB identified in knee alone and there is nothing on the chest. So if you're doing, there is no precautions for people who have been identified to have TB in the knee alone. There is no chest TB. However, whenever there is any change on the lung, you want that patient to be on airborne precautions, and staff is wearing fit tested and seal checked N95s. You want to ensure that that negative pressure room has proper negative pressure room. You as ICP have a responsibility to check on that as well. In more modern settings, you will find displays in front of the rooms, and preferably your rooms will have an empty room. And when you're looking into your negative pressure rooms, you will have a room that is negatively pressurized in comparison to that anteroom, and then the anteroom that is negatively pressurized in comparison to the hallway. So you always want the air from outside of the room to be brought into that room and not the other way around. So with regards to the communicability, so this is important, Jagdeep. This is one of the important parts of TB, is that we are keeping our patients on precautions until they have completed 2 weeks of effective therapy. So how do we know it's been effective? They have to show signs and symptoms of clinical improvement. And we also collect 3 consecutive sputums, and they have to be 3 negative AFBs. They are to be collected 24 hours apart in settings where patients cannot be available to collect the 3 specimens 24 hours apart. You will sometimes find acceptable at the same day, an hour apart. Specimens are always taken before the food consumption because you want that specimen to be not affected by food and drink. So you want that a good specimen that's collected from the lung. We will be talking in section 6 about how do we obtain these specimens, and that's going to be important also for CIC. But right now, just know that it is airborne precautions. Know the difference between latent, active, and miliary TB, and also know how long your patient is to stay on precautions. And we don't isolate babies. All right, so we come to the end of this part 1. I know.
Sorry, Cornelia, just a quick question for you. Mm-hmm. I just found out recently, like 1 or 2 days ago, from my manager, there's a whole difference between IPAC and occupational health. So I don't exactly understand the extinction. Like, I think you're supposed to tell OC health like what's going on, but you don't give like information about the patient or the person, the staff member.
Yeah, so we will be dealing with that later on in that. I will touch a little bit on in outbreak section with regards to involvement of occupational health. Right now, we are only focusing on the micro piece of all of these diseases. But when you are dealing with any of the communicable diseases, IPAC and OC health have to communicate. So we are the ones that see the patients and we know what the patient has. So, for example, if I have a patient that presented with TB, I have to follow up with a report. And it's so a lot of these diseases that we went through are reportable. TB is one of them. And you're reporting this to public health, but you're also reporting this internally to your occupational health for the follow-up with the staff. So it's important to include them. As I said, I have promised you guys I will upload these forms that I used in the past that you can also, if you want to use them. But it is to help you understand how this reporting process goes. So you want to know where your patient was, what time the patient presented, and what was the time that they had absolutely no precautions. So nowadays, most of our patients are presenting with masks, thank goodness. But before COVID, you would have somebody that is coming in and they're not wearing any mask and sitting in a waiting area for extended period of time. The good part of it for TB in particular is that you really need to have an extended exposure. So we are talking about cumulative hours, about 60 hours of being in the same room with a person with active TB. And this, when I say 60 hours, I'm not talking intubation. I'm just talking like regular being in the same room with a person. Once you start doing any of the intubations or manipulating airways, or where you're inducing the sputums, that time for exposure, of course, is shortened because the risk of exposure is heightened. We will touch on that. We will talk about that in a more detail in later sections. But absolutely, whenever you're dealing with any communicable disease, like your group A strep is reportable, you have to make sure you follow up with Doc Health. Influenza would be another one. Chicken pox would be another one. Measles would be another one. So every time you're dealing with any of the communicable diseases, you have to talk to occupational health. You just don't say, oh, we have this and it's something that we're dealing as IPAC. You always have to keep these lines of communication open with the rest of the department.
And I think that there's like certain, like, I don't know, there's certain aspects you can't like talk about an employee identifier, like no other things. It's just talking about.
It's a privacy.
You're going to talk about this later, so that's
yeah.
fine. I just want to know.
Issue. Yeah. So you as IPAC, you don't disclose the name of the patient necessarily. I mean, a lot of, depending on what you're dealing with, and most people know who they dealt with and what their patients have, and it's part of circle of care. But it's not the same when you're following up with the staff. And it could be, actually, the tables could be turned. You could actually have a staff that presents with the disease and they're coming to work, and you can't know who they are. So it becomes a little bit of a challenge, sharing that information and maintaining the privacy. And if you are in a possession of that information, you have to be very, very careful not to disclose that. Burnett, you survived?
No, I've been here. What are you talking about?
You survived?
I've been here.
She said you survived. She didn't say you left. She said, did you survive?
Oh. Oh yeah, I've survived. No, I just wanted to say that I used to chair the joint health and safety, in my community. So, number of staff with anything. We don't really need to know their names or a lot of information about them. We just need to know the numbers so that we can present that.
Exactly. So it becomes really, really interesting discussion between IPAC and occupational health. And sometimes it just can't be helped because either we as IPAC have witnessed something and simply you were there, a part of that. But the goal is not to share the identifiers from IPAC to occupational health and from them to us. So you always want to protect the privacy of whoever you're dealing with, whether it's a patient in one end or the staff in the other end. So I just want to check on you guys. Is there any questions that you would like me to go back before we wrap up tonight? Is there something that you would like me to? Okay, sure.
Cornelia, for me, I would definitely love it if you were able to just, I know you mentioned a lot of it during the slides, like which ones are very important for us to know. Like if you were just able to list it. Like for the CIC exam, like, you know how you kept on saying like TB is very important. You need to know this. Just a list.
I have uploaded on the page, I don't know if you had an opportunity to visit, a full breakdown of important and topics that they will focus on on your quizzes, on your exam exactly. So I don't know if you had a chance to look at that. I don't know that I actually.
No, I didn't, but I will look into it. Thank you very much, because I didn't know that it was there.
No, yeah, so I uploaded it last Friday or Saturday. So if you go in, let me see where I put it actually, so I can show you. Okay, so for the PowerPoint presentation, there is a question while I'm looking here. So I have sent you the doodle to pick your topics. I think about 17 of you have picked the topic before tonight's class. I have doubled some of the topics to give you the opportunity to, if it so ends up that someone else picked your favorite topic, you still have that opportunity to talk about it. So what it is, so I also have posted about the PowerPoint presentation in terms of what are the expectations from you when you're making this presentation for us. So it's on the main page where you go under the course outline. There is where I broke down your assessments and then your PowerPoint presentation, 25% of mark. When you open that, it actually speaks about what are you expected to prepare for this particular presentation. It is to be submitted at the end of the week 10. And I will be giving you 10 minutes to present. And we will break it over the course of these future 6 weeks after that, so we don't get all overwhelmed, to give you all opportunity to present your material. But you have to send it to me by the week 10. And then after you're done with submission, there is no more correction. So everyone has a fair opportunity to submit the material. And then we are going to share what you guys learned. And I think it's not going to be a nice refresher for everyone to go back to that, whatever we touched on at some of the classes. So it is end of the week 10 to be submitted. And then I'm going to create another calendar for you guys to pick your days when you want to present. And then you're all going to have a chance to present your material. So yes, 6 or more weeks. I think it's fair time that I gave you to pick your topics and start working on it. If you have any questions, you can always ask me. I'm happy to help you through that. And then I think for your question about the CIC, it is right below the course outline. I have entered another discussion topic. It's called CIC certification, and it does speak about your CIC requirements. I think someone asked me last week about this assistant ICP. There is a little bit about that. And then I put some of your CIC sample questions so you can see what can you expect to be faced with. I will also tell you that I'm creating your quiz in the same fashion as the CIC exam are. So you will be prepared for that and have an opportunity to write a similar exam with us.
I'm sorry.
Yeah, I did.
Sorry, go ahead, Burnett.
No. I was just saying that I did saw the link and the choices, and it's okay because one person or two person can have the same topic. It doesn't matter, exactly. Right? So yeah. So I have given for each topic at least 2 people can sign up for the same topic. Some of the topics that I know are usually preferred, I actually have given 4 options. So 4 people can sign up for similar or same topics. So I didn't say so Mr. Clear. Yep.
I'm sorry, Cornelia. Just about the time you were answering my question, my connection went off, so I did not hear anything of your reply. I know I saw what you said, what you're expecting for the PowerPoint. That's just general PowerPoint presentation requirements. What I'm requesting to know is, the topics are broad. You have a topic on TB, on measles. What do you want to talk about? You want to talk about the history of TB, the pathogenesis, the causative agent. How exactly how far out do you want us to go on each topic?
I want you to stick around IPOC and these diseases, or anything that we have listed, anything that is related to your practice as infection control practitioner, right? So how does MRSA how would you approach MRSA, for example, if you were to present to your colleagues that are seeing an increased number on their unit? Or if you're talking about the hand hygiene, again, from the infection control perspective, because we are learning and in process to become certified practitioners, so all our topics will always revolve around infection control. We can touch a little bit on some of the fun stuff and maybe put something with regards to those like interesting historic events, but you always want to focus on it from the IPAC perspective.
Okay, thank you.
No problem.
I had to ask you, like, I don't know where you like posted this.
Oh,
I'm trying to like find it and I don't see it.
okay. So,
Yeah, this posting also comes a little bit confusing many times. I even sent you a mail. I don't know if you saw it, because I'm not sure if it's the correct. I think it should have been correct address I sent it to, but I didn't get a reply from you, so I was wondering why. Because it's as though I get to my account today. I try to follow up with what is there. And then I go the next day, I see that something was posted even before the previous day I went there, and I didn't see it on the day I was there. So I'm wondering, there's always a lot of changes on the account that makes it a little bit frustrating to follow along with the class online, with the course online, I should say.
So when you land on your IPAC page, you will have always your general landing page. And that's where I have nowhere else to put your forums and topics for discussion. So they're broken down by the units as we are going through. And then if you're looking at the particular, so somebody at the beginning asked for a course outline, and I kept that on the top because I think that's where it's important for you to see what's happening and what's upcoming. And then your CIC certification is right under. So I don't know if I can, let me see. Okay, so do you see where we are? So your course outline is on the top. And then here is your link to your choice of your PowerPoint presentation topics. And then here is a discussion with regards to what you need to achieve in order to get your 25% of the mark.
Sorry, is that what you said? Have you picked your PowerPoint topic? Is that what you're saying?
So no, so go back, and it's right below it is PowerPoint presentation, right? You see?
Oh, okay. The second link. Yeah.
So in there is broken down in terms of some basic requirements, what you need to do to get to create a successful the presentation. So it has to be submitted by the end of the week 10. So if you're getting submissions, after that, so we are not meeting that minimum criteria, right? So you have to be.
But there's no topic, right? There's no actual topic.
No, not there. So your topics are actually in here. So I actually have a link to Doodle there. So you see this Doodle link here?
Oh, let me rewind.
Okay, so you click on that, and now you will see. So there are your topics on the top. See?
1 second, I'm just opening it up, and it just says add a new discussion topic, and I don't get any information on topic.
Okay, so
hold on. Let me try this.
Don't click in that blue thing. Go above that little blue square. You see the link? It says Doodle.
Yeah, I'm gonna try that. Okay, now I see it. Yeah, now I see it.
Right. So then you just put your name in there and pick a topic you like.
So any topic is good with you. Do you want AP style? Do you like AMA
So,
style?
So we have to follow the APA style. That's what
APA, okay. So even under like what you're showing us, I just see like names.
And then on the top, see, on the top are the topics.
Oh, okay. Thank you, because I didn't know.
No problem. I know it's like I'm trying to simplify it, and it seems that it's just getting more complicated every time I try.
Oh no, it makes more sense now. Like even if you click on that little blue button, you still have to use the link above and open it, and then you can sign up for something. So that makes more sense. Thank you.
Yeah, no problem. And then you were asking me also about your CIC post. So if you go back to the main page, so there is your, see that CIC certification?
Yes, I see it now.
Okay, so there is your CIC requirements. So I listed down there all the requirements that are needed for you guys and the topics.
So extensive, Cornelia. And you took off your headband again, and you're telling me about all these requirements.
Here. Okay, so
it makes the requirements a little bit more fine.
Okay, so now you see how it's broken down in terms of the topics and what areas are going to be bringing you the points and how many questions you will be asked from the certain areas. So you see that when you get into epidemiology, that's quite heavy.
This is APAC book, right? So it will have all this info on there. Okay.
Absolutely. And I also
have week 4, so we have 6 more weeks to create a PowerPoint presentation and submit it.
We have 6 weeks, for that.
And is there an actual email address? Because I tried to ask Leonard for an email address to send to you because the first week I wasn't around. And then the second week I had an issue with like documenting on the discussion post.
Yeah.
So I don't know if you have your, like, if you could send it to me, that'd be great, or to us. And then that way, in case you can send the PowerPoint there, because I don't know where it is.
It's also on that page, on the main page, actually. Where are we here?
Perfect. Thanks. Honestly, I just haven't had time to look.
And I know. It's okay.
Else. Thank you.
Oh, no, sorry. I gave you just a wrong address. Oh my gosh. Never mind that.
Cornelia? Yeah?
Yeah,
I have a quick question. So I did go under those topics for a presentation, and I do see healthcare associated infections, right? So I know obviously there's a few types. If I were to focus on one, such as like a bloodstream infections, would I have to specify that somewhere, or it doesn't matter?
Just say, I'm going to talk about healthcare associated infections, and I'm going to speak in this particular case about the bloodstream infection and whatever is that you picked there, right? So okay. If that's what you want to do, like, I'm really not trying to be too prescriptive. I want to give you more liberties to pick and choose here what you want to write about, as long as we're focusing on the what are the causative agents, what is the epidemiological links, what are the precautionary measures that we are putting in place, if there is a treatment, what type of treatment is there, and in terms of, again, the isolation, PPE, and more of that type of discussion. So always think about you're teaching this to the troops, to your frontline colleagues, right? So the idea is you want to use these materials in the future when you need it to teach to your frontline colleagues.
Right. That makes sense. Because currently, I mean, I cover the ICU and surgical program, and I do their like BAPS and CLI surveillance, right? So I might relate to that topic more. So let's see. Thank you.
Any topic you pick, it's just like what I'm saying is always think about who is your audience, because your presentation is not necessarily meant to teach us. Of course, we should benefit from it, but you always want to focus with your role of a practitioner. How would you apply this knowledge?
Gotcha. Gotcha.
Okay.
Yes. Thank you. Yes.
All right. So, and then back to this CIC. So you have your CIC example questions here. So I think this will give you a good idea of, I didn't set it up purposely as actual quiz because I didn't want anyone to start sweating bullets, but just so you see how does this look, what can you expect, what type of questions would there be, and I think it's a good presentation of the topics that you would see across the board. So be prepared for a lot of EPI questions. There's going to be a lot of EPI questions on your exam. There's going to be quite a lot of your micro questions as well, but infection control sort of a thing is also going to be.
EPI questions? Do you mean micro? Like what
No,
we're going to say?
but under epidemiology, what it falls, it's mostly your statistics, actually.
Oh, okay. I didn't know that. All right,
Yeah.
thank you.
No problem. So, we were going to be talking about your 2x2 tables and error 1 and error 2 and standard deviations and your rates and that sort of a thing that is linking to epidemiology, and then also some of the outbreak management and recognition of outbreaks, and that sort of a thing would fall under your EPI. So identifying and controlling outbreaks. So what else do we have here? There you go.
Is this the CIC exam you're talking about, or for our exam that we're going to have as a midterm?
So this, what I'm showing you, is for your CIC exam. Our own exam is I'm building it to be similar and replicate somewhat the CIC. I can't really make a copy of CIC without getting in trouble or sharing too much of specifics, but what is there I can share with you, what's available to anyone. So, and I do that. So if you can see here, you have your cleaning, sterilization, and disinfection is going to be about 15 questions. Environment of care is 14 questions. So what I want you to also take away from this is that not every question is weighted the same. So some of the questions will be more complex in nature and they will be worth more. So some of these questions that usually are weighted more are about your surgical site infections, usually that stratification of clean or clean contaminated and contaminated wounds, and some of that antibiotic prescription prior as in terms of prophylaxis. So some of those questions they're just simply worth more than if you're talking about, say, hand hygiene or so. Not that that's not important, but when they're comparing the weight of the questions, they're not one for one, if that makes sense.
Yes, it does. That makes sense.
Thank you, Anita. And so that's on with that regards. I also put a link on top of, if you want to go and take a look for yourself and a little bit more, there's the handbook that you can take a look at. And what else did I put here for you? So that, and then there's that assistant ICP, and I also have put here some of in the last week, you guys were asking me about some of the links that we were looking for. So there is that. And I kind of struggle where to put things for you guys because it's not really giving me a lot of opportunities to post in some different way. Then we have to pick these major headings and then I can post in there. So I could maybe create something like an ongoing kind of dumping ground for all the topics, but I kind of think it's better if we stick to the week that we discuss topics and then I leave it there for you. Oh yeah, Sharon, yeah, you can. Don't worry about it. Just go ahead and post about the SWOT analysis.
Oh, Cornelia.
Oh, sorry. Go ahead, Burnett. Go ahead.
We're always
apparently we're on the same side at the same time.
How often do they change the APIC book? Because my girlfriend just finished writing this year, and she said they might be changing the book again.
So when I spoken to them, what they have told me is they decided to go electronic because they want to keep their book fresh and regularly updated. That said, usually when you go into, if you have electronic access, they will tell you there is a new chapter or updated chapter. So one of the new chapters that I have noticed is the antimicrobial stewardship and COVID, of course. So those are the new chapters that I've seen since I had a book. There is a lot of these chapters that they go in and refresh and add information as the information becomes available and they're trying to keep current. So I really don't have any sort of an answer in timelines for you, but it's just something that I noticed that they do update on a regular basis on the electronic side, and there's no more print.
So do you have to register and become a member in order to have that access?
Yeah, so the book is members, or you don't have to be a member of APIC, but you have to pay to use the book. So if you're a member of APIC, the electronic access is about $165, if I remember correctly, and it's about $200 if you're not. The problem with all of these electronic things is that they're only available to us. It's an annual kind of subscription. So once that goes, you're kind of stuck there without having access to that. The actual book itself, the electronic version, you can always download and save it in PDF, but that kind of puts you in that at risk, that you're not going to have always the freshest information.
Updates, yeah.
So Cornelia, my question was basically like, okay, I'm not talking about the CIC. I'm talking about us in our IPAC course. Are the PowerPoint presentations, studying those, are those sufficient enough for us to write our midterm?
No.
Okay, so is there like...
to write your midterm, yes. To write your CIC, no.
Okay, so for a midterm, as long as I'm studying these PowerPoints, I should be okay.
Oh yeah,
but
for sure.
I see I need to get the APAC book and obviously do all this other external study.
Absolutely. I will not ask you in exam anything that I didn't cover.
Okay, sounds good. Thank you for that clarification, because I was sitting there going like, oh my God, I have to study all this before this midterm. Okay, thank you.
No, that would be so unfair. I would never do that. I would never do that. That's not fair. So what I teach, what I present, that's what's going to be on the on your midterm.
Thank you for the clarification.
Yeah, no problem. Yeah, so Sharon, you're right. Most organizations sign up for access to APIC books. I actually never worked anywhere that they didn't have access, or I don't know anyone that doesn't. So you can get it through work. I don't know if, Burnett, where do you work? You work for a long-term care facility somewhere, but I don't know where.
I work in the retirement with Sienna, so I'm not quite sure they have
it. Yeah. So Cam should have granted you access to that.
Okay. Okay.
Yeah, so ask Cam. Cam probably would be the best person for you to know.
Okay.
So about the EPIC book, is there a restriction as to the number of times you can download the textbook if you have access to it?
I never heard that there is a limitation. So as far as you don't really, you don't download per se. You can only access the book, and then you can print to PDF, but you can't really download it's not really coming out nicely looking when you do save it as a PDF.
Did you say print to PDF?
Yeah, you can print to PDF, but it's not the most appealing looking page of paper, right? So, but I don't think there is a limitation. Never heard that there is a limitation to that.
Okay, thank you very much.
Yeah, no problem. Oh, this one I got, Sharon, I got that from a friend of mine because I don't know, I feel like I'm a poop aficionado. I have a bunch of different poo items around my house. I have Christmas tree decorations for poo, I have pillows, I have keychain. So every time somebody sees one of those poo emoji things, when they see me next time, they bring me more. So tomorrow we'll have coffee and I will drink from my poo coffee mug. Yeah.
Oh, that's so cookie.
Okay, so
Oh,
just in case
a nice thing to end the night. Nice to end the night with that, Cornelia.
So I'll see you guys tomorrow morning at 10:30 Toronto time, and then we will continue with this exciting micro. And I hope I don't disconnect myself and do all the crazy stuff that I done today.
Thank you. Good night.
Thank
Good night,
you.
all.
Okay, bye.